here to give you a tutorial about meta-learning for bridging labeled and unlabeled data in biomedicine. The tutorial will be given by Maria Brbic, who is a postdoc in my group at Stanford, and Professor Chelsea Finn, who is one of the key figures, key innovators in meta-learning. She's an assistant professor in computer science department at Stanford University. Before we begin, I would like to say that we will be using the Zoom chat feature for questions. So if you have a question, just type it into the chat. We are going to monitor the chat and then answer your question live. The tutorial is also being recorded so that we have a digital footprint. To start, so welcome again, everyone. This is a tutorial on meta-learning for bridging labeled and unlabeled data in biomedicine, and we are very excited for everyone who's attending. The important part of the tutorial is that we have a tutorial website at the following URL. You can find all the tutorial materials, slides, pointers to papers, and pointers to tools and code at the following website. We also put the website URL in the Zoom chat. And as I said, questions can be asked through the Zoom chat feature. We are going to repeat the question live and answer it. We have a tutorial with the 15-minute break in between. I'm going to start, and then my collaborators are going to take over for other parts of the tutorial. So traditionally in machine learning, the assumption is that we have there is availability of large-scale labeled training data. Right, so the steps and how we think of the machine learning process is that first we collect a labeled data set. Right, it means we collect a set of let's say images together with their labels, and this is the first step in the machine learning process. And now that we have a set of inputs and a set of outputs, so a set of, let's say, images and a set of their labels, we then train a complex machine learning model. Today, those models are mainly being deep neural networks with a large number of parameters to basically take the input and predict the output. And this is kind of a standard supervised machine learning paradigm. But if we want to translate this or use this paradigm in biomedicine, there is a lot of challenges. First and most important is that finding large-scale labeled data set is very difficult to obtain, right? So the example I want to illustrate, right, it would mean that, for example, if I want to classify patients, I need a large number of patients, and then I need each patient to be classified, let's say, with their disease state, and I need each possible disease state so that the model is able to learn, let's say, to identify the disease from the patient data, right? And there is many reasons why this is hard, right? First is that expert knowledge is required to annotate data sets. We have incomplete understanding of biomedicine, of health, of complex biological mechanisms. So even annotating a data set ahead of time with all the labels might be impossible. And just obtaining the raw data might be very expensive because we have to do experiments to obtain the data. And naturally, right, it might be hard or impossible to even obtain enough samples, enough examples of rare cases or conditions, meaning creating such data, such completely labeled data sets is nearly impossible in practice. Right, so we have many rare and novel conditions or states in our data. So as I said, maybe in the training data, if we are thinking about cell type identification, where we are given gene expression profiles of different cells, and we'd like to identify the types of the cells we are seeing, then maybe in the training data, we are able to label, a human expert labeled identified T cells and B cells, but maybe in the test data, there is some unseen cell type that we haven't seen yet, and we don't want to just say, oh, all cells can either be B or T. There might be some unknown cell types at the deployment time. right? So we can have never before seen classes that appear at the test time or at the deployment time. And this is very important because it's necessary, right? The goal of these models is to discover new biology, like new drugs, new diseases, new cell types. So they need to be able to learn in the presence of unlabeled or partially labeled data, and in the presence of novel types, classes that were never seen or discovered before. Another challenge is that learning in biomedicine requires generalization across diverse biological contexts. For example, we want to be able to learn across species, right? Maybe we have data for certain organisms, and now we'd like to use that data to learn a model that is going to work or be able to require less data for a given new organism that hasn't been studied or labeled before. Another example of this kind of generalization is to generalize across tissues, right? Maybe we have labeled data for brain, heart, and lung, but we would like to generalize to unlabeled data that we get, I don't know, from the colon or from liver or some other human organ. So this is hard and very hard to do with traditional machine learning methods, right? So we want to generalize across species, across tissues, across cohorts, diseases, and time points. So how do we deal with this type of complex data, missing labels, and with this goal to generalize to unseen domains, to unlabeled tissues, unlabeled brand new organisms that are, for example, studied for the first time? And the paradigm that allows us to do this, we are going to talk about them today. First, we are going to talk what we call few shot learning, where the idea is that we want to predict correct classes given a very small number of labeled examples in the training set. And I'm going to give an example about this, and it's really remarkable what is possible to be done today with these types of methods. For example, what do I mean by having a small number of labeled examples? It could be maybe we only have one case of a very rare condition. We maybe only have a few cases of a very rare cell type, but we'd like to learn from only a few cases to be able to identify other instances of that type. And then another part that we are going to talk about today is what we call open world learning, where in machine learning, generally the assumption is that it's a closed world assumption, right? That all the classes have been labeled, predetermined ahead of time, right? So the idea is if I'm building a species classification engine, then at the training time, I'm going to see all the species, all the possible species ahead of time. And like usually, right, if a brand new species appears in the test data at the deployment time, such models wouldn't even be able to say, oh, this is a species I've never seen before. They would just try to fit it into those predetermined set of classes they were given at the beginning. So the goal of open world learning is to predict correct classes without any examples belonging to that class in the training set. So basically to be able to identify a novel class, a novel instance never seen before. And if we think about biological discovery, this is the core, right? Is we want to discover a new class, a new condition that has never been seen before, that is not part of our labeled training data, hasn't yet been even recognized or described before. So rare conditions, identification of rare novel cell types is an example of these types of tasks. So just to give you an example, for example, a few shot learning approaches can be used for protein function prediction, where a large number of functional labels may only have a few genes annotated, right? And we have a very little training data for each functional label. And the task here would be to assign genes to functional groups, where we only have a couple of examples of genes for each functional groups. This is hard to do because simple models won't be able to generalize well. They will have high bias and low variance, and the complexity of the data is also very large in terms of having noisy labels with large numbers of features. And just for traditional machine learning, there is not enough annotations, there is not enough label data to train highly expressive models using traditional approaches, right? An example of open world learning is that perhaps we want to discover a new disease, maybe a new type of disease. We know everything here about the flu, but then imagine a new disease appears, let's say COVID-19 or SARS-CoV-2, where we want to learn something that has some other different properties, and the model needs to be able to recognize, to say, oh, this is something new. I've never seen this type of disease, this type of virus before, and here is how it differs from previously seen examples from which the algorithm originally learned. So the task here is, right, to assign, let's say, patients to this novel disease, to the disease states, existing ones, as well as being able to identify a brand new disease state. Why is discovering novel disease states hard? It's because we have no labeled examples of the novel case, because it hasn't yet been discovered before. It is much more complex than outlier detection, in a sense that we want to identify examples of unseen cases, and then group similar examples together to say, oh, this is a new class, and here are all the instances of that class, rather than just say, here is a big group of outliers, we don't know what these are. We want to also identify coherent groups of them and proclaim them as new classes, as new types that we have never seen, or the algorithm has never seen before. So this tutorial is all about what do we do when this assumption of availability of large-scale labeled data is violated, right? What can we do when we have small, partially labeled data sets that are very different from each other? How do we effectively learn from them? And if we think about the problems in biomedicine and experimental natural sciences, it's a prototypical example of this, because data is collected by experiments, and the same experiments are not repeated over and over again, because they are usually done to discover something new that hasn't yet been discovered or labeled before. So every new experiment generates a new data set that is different conceptually from other data sets that have been generated before, right? There is a new condition, something is different in that experiment we are trying to do. So machine learning methods need to be able to learn with such rich data. So to give you an example of this and how remarkable it can be, imagine I'm trying to identify species of birds. And here I have 5 different species of birds, and imagine that I give to the learning algorithm only a single image, a single instance for each of the species. I say crow, here's one instance of crow, here is one instance of raven, here is one image of a nutcracker, and so on. And the question is, can a learning algorithm learn from just a single image of a raven to be able to identify all kinds of ravens in all kinds of backgrounds, in all kinds of shapes and positions? Right? So at the test data, I'm given a huge number of images, and I'm asked to assign species to images, where at the training time, I only see one image, one instance of each of these species. This is called one-shot learning, because we get only one training example per class. And what is remarkable is that these types of methods that we are going to learn about today are able to identify species with 70% accuracy, even if we only have one training example per species. And that's quite amazing if you think about it. So that's for the motivation of the tutorial. And we are going to talk about how are we able to achieve this type of performance given so little training data, given only a single instance of the training data. So if you wonder how, you should watch this tutorial. Right, so this tutorial will be about how do we learn from very few labeled examples. How can we learn to transfer knowledge across different biological contexts, meaning between different diseases, between different species, between different tissues? How can we generalize to novel, never before seen classes? How do we identify rare and unseen cell types, diseases, and similar? So this is what this tutorial will be about. So if you wonder how to answer these questions, you should stay tuned. The structure of our tutorial can be organized in the following way. we are going to split the missing data problems into two buckets, into the few shot learning bucket, into the open world learning part. We are then going to talk about the methods here in light green. So from optimization-based methods, non-parametric machine learning methods, methods that incorporate prior knowledge, and then also methods about novel class discovery and open world sampling supervised learning. I've just walked through the introduction for the first 15 minutes. We are then going to talk about the few shot learning. Chelsea is going to cover this part for 90 minutes. Then we will do a 15-minute break. And then we will start with part 2, which will talk about prior methods that can be enhanced by prior knowledge. And in biomedicine, we have a lot of prior knowledge captured by various data sets, ontologies, and so on. And then for the last part of the tutorial, we are going to talk about open world learning. And in the end, we are going to talk about some practical cases, discuss the tools, discuss how you can do these things by yourself, and conclude. And that's the agenda for today. So what I want to do next is very briefly bring everyone to the same point and give a brief overview. How do we think as machine learners, and how do we think especially in deep learning? So I want to introduce some basic concepts so that we'll be on the same page for the rest of the tutorial. So in machine learning, we think of machine learning as optimization, right? And in particular, in supervised learning, we are given a set of inputs X. This could be, let's say, images. And our goal is to predict a label of each input. And we are going to use Y for the label. So I could say I'm given an image X, or an image of the bird, and Y is the species of that bird, right? So the input can be a vector of real numbers, like a gene expression profile. It could be a sequence, like a natural language description of something. It could be a matrix, so a matrix of numbers. So an image can be represented as a matrix or as a tensor of numbers. Or input X could even be a graph. For example, a molecule can be represented as a graph, as a set of bonds between the atoms. So that's the input. And then the task is to predict some property, some label of each of these inputs. For example, for molecules, it would be whether the molecule is toxic or not. Right? And now we want to learn a function that will take X and output Y. And learning this function will be formulated as an optimization problem. So we want to formulate the task of learning the model, the function F, as a optimization problem. The point being is that we are going to define what is called a loss function L that takes in the inputs, the input labels Y, as well as the input data X. And our goal will be to learn this function F that is parameterized, described by a set of parameters theta. And our goal will be to find the parameters that minimize the loss. And the loss quantifies the discrepancy between the prediction f of x and the truth y, right? So for example, if we would be predicting some real value, then you could define loss as a L2 distance, as a square root of the square of the distance between the true value y and the predicted value f of x, right? And then the goal is to find the parameters of f, to find the machine learning model f that minimizes this loss, that minimizes the discrepancy between the predicted value and the ground truth value. This is for, let's say, simple regression tasks. The common loss that people use for classification tasks is what is called a cross entropy loss. So for example, if we have a 5-way classification type, we have, let's say, five types of birds to classify, we can represent the label as what is called a one-hot encoding, right? So 0 means that a given bird is not of that given type. So this particular bird would be of species number 3 because it's a 1 at the position number 3. And then we can ask our function f of x to return us a 5-dimensional vector where each value will tell us what is the confidence, what is the probability that a given species, that a given image is of a given species, right? And here we could see that the highest probability is assigned to the class number 3, to the species number 3. So we would now measure the discrepancy between the, right, what we would want is for our classifier to assign a very high probability to the third entry of this vector. And we can write out the cross entropy loss as the following, with the following equation, where y is the entry of this vector and f of x is this probability, and we take a log of it. Log is a monotone function. So basically the idea is to put small probabilities to the entries of y that have 0, and we want to put a high value, a high probability, high f of x to the correct entry that has the entry 1 on the vector y. And this is called a cross entropy loss. Right? So now we have an optimization problem, and we want to find a model that minimizes the loss, that minimizes the discrepancy between the predicted label and the true label. And these optimization problems are solved with gradient-based methods. So kind of one of the most basic intuitions about how do you optimize a function. You optimize a function that you define, you compute a derivative of a given function. You evaluate the derivative or at a given point. Here a point means theta, and then you update the theta with a small step in the direction opposite of the gradient. And this parameter here is the learning rate. It tells you what is the step size in your parameter update. So at the training time, we are going to optimize this theta, the model parameters, iteratively. In one iteration, we are going to do one step of gradient descent. And the problem with traditional gradient descent is that computing gradient requires evaluating the derivatives of all the data points in your training data set. And if the training data set is large, then this might be very expensive. So what is done in practice is what is called stochastic gradient descent, where at every iteration, we pick a small subset of the training data. We call this subset a mini batch. We evaluate our gradient of the loss only on that subset of the training data on the mini batch and update the parameters. The benefit is that now we evaluate the gradient much faster, but the drawback is now that the gradient is only approximate because we only evaluated it on a subset of training data. But in practice, this works much better because each step is much faster because we only have to consider the mini batch of data. So the important concepts here are batch size, which is the number of data points training examples in the mini batch. Now, one iteration of stochastic gradient descent is processing one mini batch, and then an epoch is a full pass over the data set. So processing all the mini batches from the data set, that is called one epoch of training. And usually we would do multiple epochs of training, which means we traverse the training data set multiple times. And then, based on this simple intuition, there are many optimizers like Adam and Adagrad and so on that basically are thinking about how do you correct the noise in the gradient, how do you decide the step size, and this is kind of where the innovation comes from. But the basic intuition is as I explained. Now, what remains unanswered is what is this function f? What is the machine learning model? And this function f can be a simple, let's say, linear layer, a multi-layer perceptron, so a multi-layer neural network, or some other type of neural network, like a recurrent neural network or a gated neural network, right? A linear layer is simply defined by saying the output at a given layer is some parameter matrix W times the input vector plus the bias term B. So here W and B are our model parameters. So theta equals matrix W and the bias term B, right? So that's the idea, and we want to learn these two parameters. The function f can be more complex. These single layer perceptrons can be stacked together and by adding a non-linearity between them. So here L is the layer number. And for example, now a 2-layer or a 3-layer neural network would take, let's say, a 2-dimensional input and then process this through a series of transformations of these WL and BL parameters that can be learned all the way to the final output, which depends on the task we are trying to do. So to summarize, we can think of machine learning as an optimization problem. We are having a loss function, and we have a simple mini batch of inputs. We evaluate the gradient of the loss at a given set of parameters over the training data, and then we update the parameters with the goal to minimize the loss. And function f can be any neural network which we can differentiate and obtain the gradient. So this concludes the introduction, and now I will give the word to Chelsea to start talking about more advanced topics. Thank you, Chelsea.
Great. And as a reminder, feel free to put questions into the chat as we go along. So we've kind of gone through the kind of introduction part, and now we're going to start talking about few-shot learning. So what does few-shot mean here? Few-shot means that our training data consists of a very small number of labeled examples. This can be as small as just like 1 or 2 or a few examples per class. And we saw the example that Jure gave before, where we want to learn to recognize bird species with just a single example per class. So how do we actually go about doing this? Well, in standard supervised learning, the way that this would work is you take your training data and then train it to predict magpie for the corresponding image, Jay for the corresponding image, and so on and so forth. And unfortunately, in standard supervised learning, if we try to train a network with just 5 labeled examples, it's going to severely overfit. It won't be able to then, it'll be able to classify these as 5 examples, but it won't be able to classify new examples of jays, crows, ravens, and so forth. So how do we actually go about doing this? Well, this is actually a problem that humans can solve. And so if I gave you these 5 training data points, and then I asked you, which bird is this one? Is this a jay, a nutcracker, crow, raven, or magpie? You'd probably be able to tell me that this is a crow. And you'd probably be able to tell me that even if you haven't seen these particular birds before or aren't really an expert on this problem. And the way that you can do that is that you have previous experience. You aren't completely learning from scratch tabula rasa, and you can leverage previous experience looking at images, looking at different animals to recognize distinguishing features from these birds with just 1 example per bird. So with this in mind, the goal of metalearning is to allow neural networks not to learn from scratch and to instead allow them to leverage previous experience when trying to learn from very small data sets. So the key principle here is, in particular, to use previous experience of related tasks to generalize to new tasks with sparsely labeled data. So for example, say we want to generalize to rare conditions or rare cell types, and we want to be able to learn predictors from only a few examples of those conditions or cell types. What we might try to do is take data from other conditions and other cell types that are much more common, learn how to recognize those conditions and cell types, and use that previous experience when trying to identify more rare conditions and cell types where you don't have a lot of data. And so this is your previous experience when you have abundant labels, and the rare conditions are the few-shot learning problem. Now, what do I mean when I say that we're trying to generalize to a new task from previous tasks? Informally, a task is essentially taking a data set and using that to learn a model using some loss function. And so in particular, we say we have this neural network, we have an input X, and this could be something like a gene expression vector, an image, or something else. And we want to be able to predict the corresponding label. And the essentially and these labels could be things like different conditions and so forth. And this neural network here is the model, and it has parameters that we're going to denote as theta. Those are essentially just the weights of the neural network. So what a task corresponds to is in single task learning, single task supervised learning, we typically assume that we have some data set which has input-output pairs. It has pairs of X's and Y's, and we want to minimize some objective. And the process of minimizing this objective corresponds to the task of learning that predictor, learning that function. Okay, and so for example, a typical loss might be to minimize the negative log likelihood of your data. So this is kind of informally what a task is. More formally, you can also think of it as essentially corresponding to the data generating distributions over your inputs X and your label given the X, as well as some loss function. And then for each of these tasks, you would have some corresponding data sets. And I can actually break down your data set into a train set and a test set, and assume that you have a train and test set for every single task. So for example, having a train and test set for every single condition or every single cell type. So what do these tasks actually correspond to? So let's look at an example where we want to associate patients to different cancer cell types. And so different tasks are going to correspond to the prediction of cancer subtypes across different cancers. So different tasks will vary in the in which cancer you are looking at. So for example, one task might be to predict the subtype of different of different forms of renal cancer. Another task might correspond to breast cancer, where we want to predict if it's luminal A, luminal B, and so forth. Another task could be bladder cancer, and so on and so forth. And if we have all of these tasks, then we can actually use this experience from these tasks to actually be able to predict cancer subtypes of a more rare type of cancer. And so, for example, maybe we have a new patient and we want to, we have some data of a few different subtypes for a new type of, or a rare type of cancer, then we want to be able to predict which subtype a patient has, really with only a few examples of data from that type of cancer. Okay, and of course, if you just try to kind of predict this new cancer subtype from a few labeled examples just from scratch, this is going to lead to a lot of overfitting in the single task learning framework, in the standard supervised learning framework. However, kind of the key idea is if we learn a shared model across each of these tasks and transfer that information to the new cancer subtype, then we can actually generalize to this new task with only a few labeled examples. Okay, so as another example, we could also look at the problem of cell type annotation, where different tasks correspond to predicting the cell type annotations across different tissues. And so different tasks correspond to different tissues. And then kind of our goal will be to then predict new cell types in a new tissue from a few labeled examples. And essentially handling these new cell types is exactly what metalearning allows you to do. If you have a very common cell type, then you don't need to, very common cell types from very common tissues, then you don't need to, you don't need metalearning. You can just use supervised learning. It's particularly when you have a very small amount of data in these more rare conditions that metalearning can be super helpful. Okay, so what is exactly the metalearning problem? We can formalize it as given data and experience from a previous set of tasks, we want to quickly solve a new task. And what I mean by quickly here is using a small number of labeled examples. And one really key assumption here is that the meta training tasks and the meta test task are drawn from the same overlying distribution of tasks. They need to have some shared structure, and they need to be drawn from a single distribution, because this is what allows you to generalize to a new task. If your new task is completely unrelated to all the tasks that you've seen before, or kind of just completely unrelated, then you won't be able to leverage your previous experience to very, very quickly solve this new task. Okay, so we kind of can assume that there's some distribution of tasks and that each of the tasks are drawn from that same distribution. Okay, so how is this different from standard supervised learning? So in Metalearning, we're given these previous tasks and we want to quickly solve a new task, whereas in standard supervised learning, we're given data from some task and we want to solve that same exact task. So there's just a single task and we assume that we have available, we have enough labeled data to learn this single task from scratch without any data from previous tasks. So in supervised learning, we try to generalize to new data points within the same task, whereas in Metalearning, our goal is to generalize to entirely new tasks with small amounts of data by leveraging data from previous tasks. Okay, and there's also other problems in machine learning that are related to Metalearning that I'll just briefly cover here. One is the problem of multitask learning, where we want to solve multiple tasks all at once. And essentially what this corresponds to is trying to train a single model that can solve all of the different tasks by simply summing the loss functions of each of the tasks. And this is different from Metalearning because in Metalearning, our goal is to solve a new task by leveraging data from previous tasks rather than training them all together. And then second, there's also this problem of transfer learning where we've solved some source task and we then want to solve a new target task by transferring knowledge from this previous task. In some ways, you can view this as a special case of the Metalearning problem because you have just a single previous task and then you want to solve one new task. However, by assuming that we have multiple different meta training tasks, we can actually do more. We can leverage those tasks to much more quickly solve a new task than if we just had a single task. And then lastly, in transfer learning and Metalearning, one assumption that's quite nice is we don't need access to the previous tasks at adaptation time. We can essentially discard all of the information from these previous tasks and just have our parameters that are used to quickly solve a new task. And in all of these settings, we assume that the task must share some sort of structure to allow you to actually generalize by leveraging data across tasks rather than just training single tasks in isolation. Okay, so now I mentioned that we don't want just one task. We want to have a set of previous tasks so that we can quickly learn new tasks. But how many tasks do you need in order to quickly solve a new task? This depends on the application, but one kind of general rule of thumb is the more tasks you have, the better. And this is essentially analogous to more data in machine learning. The more previous tasks you have, the more easy it will be to generalize to a new task. Okay, so that's kind of outlining the problem statement. How do Metalearning algorithms actually work? So the general strategy of these algorithms is we want to solve this few shot learning problem, similar to the bird classification problem before. Maybe instead we want to classify between ice cream, a certain breed of dog, a lion, and so forth. So we have our training data, and our goal is to generalize to new examples using these 5 examples. And the way that Metalearning works is instead of assuming that we only have these 5 examples, we instead assume that we've seen previous data of other images, of other image classes, and we want to leverage kind of tasks, different image classification tasks that we've seen before in order to solve this new image classification task with a small amount of data. And so in particular, the way that we do this is we can structure our previous data in the form of these different tasks and train a model such that once it's seen the training data shown in green, the model can then generalize to the examples shown in blue. And we perform this optimization, this kind of training the model to generalize from these examples to new examples across all of the tasks that we have, across all of these training classes, so that at adaptation time, we can then learn new image classes from just one example per class. So this is kind of the meta training process where we try to learn from each of these tasks. And then at meta test time, we're given a new task. And because we've seen all these tasks during meta training, we've prepared ourselves to do the same exact thing at test time and generalize from a few examples. Okay, and of course, this is an image classification example, but you can replace classification with a wide range of other things. You can replace these training sets and test sets with regression problems, with language generation problems, with cell type prediction problems. Really, any machine learning problem can be kind of fit into this framework. Okay, so a couple more terminology before we continue. So we're going to be calling each of these kind of green boxes the task training set. So the training set for task I, and each of the blue boxes as the test set for that task. So we have a training set and a test set. These are also sometimes referred to as the support set and the query set. And the support and query terminology can be useful to try to differentiate the train and test sets with the meta training set and the meta testing test set. Okay, and then we view the K-shot learning problem as learning with K examples per class. So in one-shot learning, we have one example per class, and two-shot learning, we have two examples per class. Or in the regression case, we assume that we have K examples total. And then we will use the kind of the letter N to denote the classification between N different classes. And so in this example, if you think about what are K and N, we can see that we're trying to classify between 5 different classes in each of these tasks. And so this means it's a 5-way classification problem, or N equals 5. And we see that in each of our training sets, we only have one example per class. And so we're doing one-shot learning. So this is referred to as 10-way one-shot classification. And K and N can be different numbers depending on your problem. Okay, so any questions before I move on to actually talking about how we go about the adaptation process?
So there is one question from Leonardi who says kind of that apart from having previous experience, we as humans are also able to extract features from single image quite nicely. For example, color, feathers, shine, and so on. So can we optimize this in the few-shot learning?
Yeah, absolutely. And when I was referring to the kind of having previous experience, like the ability to extract features, if you just learn completely from scratch, like maybe when you're a baby, you probably can't extract features as nicely. And so this meta training process is exactly what's allowing you to extract these nice features to allow you to generalize to new image classification problems.
So another question is regarding slide 20. Is meta-learning a certain way of formulating a learning problem on the same kind of inputs, like split up 1,000 classes into 100 tasks instead of doing 1,000 multi-class predictor?
Yeah, so that's one way that you can view it is essentially kind of, you can formulate, yeah, so that is like, that's correct, essentially. And if you want to be able to kind of learn classifiers, image classifiers on the same kind of image input, this allows you to do that. In principle, you could also have tasks that have different inputs, but typically you assume that you have the same sort of modality and the same kind of problem. And your goal is to kind of learn about that modality and learn features, learn kind of the structure that's shared among those tasks in a way that allows you to make a classification for a new task.
Thank you. I think we are good to go.
Great. So one way that we could approach this problem is to just try to pre-train the model on our previous tasks and initialize the model using those pre-trained weights, and then fine-tune your model on your new task. And the way that this works is you kind of pre-train some parameters of your neural network on your previous tasks. And then you run gradient descent on your training data for a new task with some step size. And so this is showing kind of one step of gradient descent, which would then give you the parameters for a new task. And typically you would run kind of multiple steps of gradient descent to fine tune your network and hopefully get a model that can effectively predict, accurately predict the correct class for your new task. And so yeah, this is typically done for many gradient steps. And this is actually a quite widely used paradigm in machine learning to leverage previous experience. And as an example, people have used this for taking language models and then fine-tuning them for text classification. And what this plot is showing is the performance on the target task as a function of the number of training examples you have for that target task. And the blue line is showing if you just train from scratch on that target task, and the green and orange lines are showing if you run fine-tuning. And so what we can see is first, the orange and green lines are significantly below the blue lines. So we get significant benefit from fine-tuning compared to training from scratch. And yeah, I'm just describing transfer learning here. However, one thing that you'll notice is that when you have fewer training examples of your target task, say 100 training examples, the performance of fine-tuning isn't nearly as good as when you have, say, thousands of examples. And so fine-tuning isn't as effective in this small data regime where you have, when your target task only has a small amount of examples. Okay, so this is where we get into meta-learning methods. And so we're going to start by talking about optimization-based meta-learning methods that allow us to perform few-shot learning and allow us to transfer to new tasks with a small amount of data. Okay, so in particular, we're going to cover the overall approach and some of the challenges and solutions with this approach. And the goal of this section will be to cover the basics of optimization-based meta-learning, including how to implement these algorithms. So the key idea is we have our set of tasks, and we have kind of training examples and test examples for each of our tasks. And what we're going to try to do is we're going to take some initial neural network parameters and run gradient descent on our training set to get some adapted parameters. And then we want these adapted parameters to generalize to new examples, even when our training set has only a small number of examples. And so what we're going to do is we're going to pass our test examples into these updated parameters and train this whole optimization process so that the predicted label on this test example is correct. So essentially training this optimization process so that we can generalize, produce a network that generalizes from a small number of examples. And in particular, what I mean by when we optimize this optimization process is we can learn these initial parameters theta. And we can also learn other things like the learning rate and other kind of parameters of this optimization process. So essentially the key idea of optimization-based meta-learning is to embed an optimization process into this inner learning process. So why might this make sense? So let's go back to fine-tuning, and we have our set of pre-trained parameters, and we want to fine-tune and run gradient descent on training data for the new task. So we'd love to be able to run fine-tuning on a small number of examples at test time, but we may not have that much data. And so the idea here is what we want to do is we want to take the fine-tuning process and train the initial parameters so that this fine-tuning process accurately predicts examples on the test set. So we're going to take the updated parameters, say after one gradient step or a few gradient steps, and optimize this loss function on new examples. So we're optimizing for an initial set of parameters such that we can generalize from this training set to this test set. And importantly, we're going to do this optimization not just over one task, but over all of the tasks that we have in our meta training set. And so essentially, over all of our meta training tasks, we're going to learn a parameter vector or an initialization that transfers well via fine-tuning. So we're learning a initialization for fine-tuning that allows fine-tuning to work with a small number of examples. Okay, so this is referred to as model agnostic metalearning, or MAML. And the reason for that is that essentially this process, this optimization, is agnostic to the model and the loss function as long as they can be amenably used with gradient descent. And so kind of visually what you can view this process looking like is this following graphic, where theta is the parameters that we're meta-learning, and phi I star is the optimal parameter vector for different tasks. And if, for example, we're at this point in the meta training process and we take a gradient step with respect to task 3, we're quite far from the optimum for task 3, and likewise for other tasks. So in the meta training process, we're going to essentially optimize for a set of parameters such that if we take a gradient step, we're very close to the optimum for that task, and likewise for all of the tasks in our meta training set. Okay, so we're optimizing for this initialization so that we can run gradient descent for lots of different tasks, and even if we're running gradient descent for a small number of examples. And so one of the insights here is that small perturbations to the parameters at this optimum right here will produce big improvements on the function when altered in the direction of the gradient. Okay, so what does this look like as an actual algorithm? So this is kind of showing the objective that we want to optimize is we want to optimize for this initialization for fine tuning. And the way that we can optimize this is first we'll sample one of our tasks, for example, one of our conditions. Then we'll sample disjoint data sets, a training set and a test set, or a support set and a query set from our data set for that task. And so, for example, maybe this is our data set for an image classification problem. And what we're going to do is we're going to just randomly split this up into a training set and a test set. So we want to kind of get some training data and some test data for this task. We'll just randomly assign images from our data set into each of these sets. And then we're going to first adapt our parameters, our current meta parameters data, using the training data right here. This will give us some task-specific parameters phi I. And then once we adapt our parameters to the task, we're then going to update our initialization using the test set. So, and this essentially corresponds to this gradient right here. So we're going to update data by taking a gradient step on the loss function with respect to these held out examples. And then we'll repeat this process and iteratively kind of resample tasks and optimize for initial set of parameters so that gradient descent and fine tuning generalizes well to new examples. Okay, so there's a couple things that you might wonder here. First, you might see that this actually requires second order derivatives. And in particular, if you look at this gradient, this is the gradient of this loss function, and this is with respect to phi I, and phi I is this expression right here. And so we end up getting a double gradient when you combine these two steps. Now, how expensive is this? Is this a problem? And do we need to compute the whole Hessian of the neural network, which would be pretty problematic because that'd be a huge matrix? And it turns out the answer to this is no. We can actually get away with only requiring Hessian vector products to compute this gradient or this expression right here. And this is something that there's lots of tools for doing this, and I guess also deep learning libraries like PyTorch and TensorFlow and so forth can do this computation for you if you simply construct the computation graph. The other thing you might wonder is, well, this is showing just one inner gradient step. What if we have many inner gradient steps? Do we get higher order derivatives, higher than second order? And the answer is also no for this. Fortunately, if you actually just keep on running multiple inner gradient steps, because the other gradient steps will not be with respect to theta, they'll be with respect to the updated theta. You won't actually get higher order derivatives, and it'll only always be a second order optimization that requires these Hessian vector products. which are much cheaper to compute than trying to construct the entire function. Okay, so that's kind of the basics of optimization-based metalearning. There are a couple of challenges that come up. The first challenge is that you have this bi-level optimization, and it can be somewhat unstable. And so there's been a number of tricks that people have developed, such as learning the inner learning rate, optimizing only a subset of the parameters in the inner loop, trying to decouple the learning rate and the batch form statistics at each gradient step, and also introducing context variables. And I don't want to necessarily go over all these in detail, but really the takeaway here is that there are a range of simple tricks that can help the optimization significantly. And if you're interested in learning more, you can take a look at some of these papers. Okay, and then another challenge that can come up is that if you have a lot of inner gradient steps, this can be relatively compute and memory intensive because you need to store the full computation graph of running that inner optimization. And there's two ideas that can actually help with this. The first idea is instead of actually storing all of those inner gradient steps and then back propagating back from your updated task parameters phi I back to theta, you can just crudely approximate this Jacobian as the identity. This is incorrect. This is not an identity matrix, but it turns out that actually if you approximate this identity, it actually surprisingly works fairly well for simple few-shot problems. And this is kind of known as first order mammal or reptile, and it corresponds to a first order optimization rather than assuming the full second order optimization. Okay, and then the second idea is instead of in the inner loop, instead of fine tuning the entire network, you could just optimize that last layer of weights. And what this will correspond to is optimizing a set of features such that a linear predictor on top of those features accurately performs the task. And so this gets back to the question that we had before, is you can actually just try to learn features such that a linear predictor on top of that can effectively solve the task. And this has been done in a number of different works as well. And you can actually, in this case, if you're just learning this linear predictor, in many cases, this will lead to either a closed form or a convex optimization on top of those features. And differentiating through that is cheaper than if you were to fine tune the entire network. Okay, so the key idea of optimization-based metalearning is to acquire these task-specific parameters through an optimization and construct this bi-level optimization problem so that you can learn from a few examples. There are a couple of benefits of this kind of approach. The first is that even at the start of metalearning, it basically just corresponds to fine-tuning or running gradient descent. And so you have a positive inductive bias at the start of metalearning, you're already going to be doing something reasonable. This also tends to extrapolate better because you're running gradient descent. And even if you have a task that is a bit different from the task that you saw before, in the worst case, it will give you something similar to fine-tuning. And in the best case, it will give you something much better than fine-tuning. It is also quite expressive if you have a sufficiently deep network. And it's also relatively easy to combine with your favorite architecture or loss function. And then the downsides of this approach is that it typically requires a second order optimization, and this can make it compute or memory intensive. Okay, any important questions before I move on to non-parametric methods?
So one question is, does this method suffer from catastrophic forgetting? If not, might this be a method for an improved transfer learning where backbones are less susceptible to catastrophic forgetting?
Yeah, so this doesn't suffer from catastrophic forgetting because essentially essentially what you do is you always, whenever you're trying to adapt to a task, you just reset to your learned initial parameters and you store those. And that kind of contains all the information from the previous tasks. And then you can adapt to your new tasks with that. The people have also used this as actually techniques for overcoming catastrophic forgetting. And so it can certainly help with that. The meta training process itself, if you, for example, have a sequence of tasks rather than a batch of tasks that you're given, then this can suffer from catastrophic forgetting, just like any other gradient descent procedure, because these set of parameters This kind of meta optimization corresponds to kind of a standard SGD problem. And so if you don't have all the tasks during this meta training process, then it will forget the tasks that you saw before.
Do we have time for more questions?
One more, yeah.
Sounds good. So I didn't understand what is the difference between meta learning and supervised learning. You still need to train your neural network with lots of data to not overfit. And the closer your meta training to meta testing, the higher accuracy you get, right?
Yeah, so you still need to train it on previous tasks, and you need to have sufficient tasks or data from those tasks in order to learn this initialization. However, once you learn this initialization and leverage this previous data, then what this allows you to do is to learn new tasks with a very small amount of data. So for example, if you have data from previous image classes and you have enough of that data, then you can actually learn a new image classification problem with many fewer examples than if you were to learn from scratch, and if you were to try to fine tune on that classification problem. Yeah, so I think that's the kind of one of the biggest differences between supervised learning and meta learning, is that you have this ability to adapt to new problems with a small number of examples once you perform this kind of optimization.
Thank you.
Great. So we've talked about optimization-based metalearning. Now we're going to talk about one more class of metalearning methods, which is called non-parametric methods. So we're right here. And so in particular, we're going to cover non-parametric and metric-based few-shot learning methods. We're going to cover 3 different methods. And the goals here will be to understand the basics of non-parametric few-shot learning techniques, the trade-offs between different metalearning methods, and some familiarity with these kind of different applied formulations of metalearning. So far we've been learning these parametric models, and in optimization-based models, the key idea was to embed this optimization inside the inner learning process. And the benefit was that the structure of optimization was embedded into the metalearning process, but that this required a second order optimization. So can we instead try to embed some sort of learning procedure without requiring the second order optimization? So the key idea of non-parametric methods is that when we're in low data regimes, non-parametric methods like nearest neighbors are quite simple and they work pretty well and they're computationally efficient. And so at meta test time, few-shot learning we're in this low data regime at meta test time. However, during meta training, we still want to be parametric because we still want to leverage our previous experience. So can we use parametric meta learners to produce effective non-parametric learners? And so what do I mean by this? So say we have our few-shot classification problem that we talked about before, where we have some training data points and a new test data point. One thing that we can do is what's called nearest neighbors. And this is a non-parametric method where we just compare our test data point to each of our training data points. And we take the one that is closest, the nearest neighbor, and take that label and predict that corresponding label for our test data point. And so, for example, if we were humans, we're kind of doing this, we compare this image to all of these images, note that it's closest to this one, and then say, okay, whatever class this corresponds to, I'm going to predict that class. Okay, so we're going to be comparing this test image with all of our training images. Now, the question that comes up is, in what space do we compare these images, and with what distance metric? And in particular, if we compare images in pixel space, then this won't actually give us what we want. So for example, if you compared this image with these 2 images in pixel space, it would actually tell you that this image is closer than this image. Pixel space L2 distance in pixel space isn't a very good metric. Likewise, in other spaces, like in gene expression space, like L1 or L2 distance isn't going to kind of accurately measure the kind of similarity between 2 images or 2 inputs. And so the key idea of these kinds of meta-learning methods is to actually learn a metric and to learn a distance function that allows us to compare the test example to each of the training examples. And so if we can learn a good metric, then we should be able to use that metric with nearest neighbors to effectively predict the correct label. And so as a first simple example of how this can work, what we can do is we can train a Siamese neural network to take as input 2 images from our meta training set and output whether or not they are from the same class. So we can train this neural network to predict 0 for these 2 images because they're different, and then predict 1 for these 2 images because they both correspond to bowls, and 0 for these 2 images because they're different classes, and so on and so forth. And then once we have this Siamese network that can essentially tell us whether 2 images are from the same class, we can then, at meta test time, compare our test image to each of the images in our training data set and use that to predict to find the correct label for our test image. And so, for example, if this is our test data point right here, we would feed these 2 examples into our network, feed kind of these 2 examples into the network, these 2 examples into the network, and so on and so forth, and find the one where the network is most confident that the 2 images are the same, and then predict the corresponding label of that training image. Okay, so this is like the simplest non-parametric meta-learning technique that is essentially learning this metric so that you can perform one-shot or few-shot classification. Now, one thing that you might note is meta-training corresponds to just this binary classification problem that we've set up. And at meta test time, we're actually doing an n-way classification by actually running the network n times for each of the pairs that we have here. And so one thing that's unfortunate here with this sort of approach is even though it's very simple, we actually have this mismatch between what's happening at meta training and what's happening at meta testing. And this means that we aren't going to be effectively preparing our model for exactly what's going to be coming at test time. And so can we match meta training and meta testing so that we're preparing it as well as we can for the n-way classification problem that we'll get at test time? And so this is the key idea behind matching networks. And the way that it works is we're going to try to do nearest neighbors in our learned embedding space and take our test example and each of our examples in our task training set. We're going to do nearest neighbors with each of these examples in some learned embedding space, and then essentially differentiate through that entire process of performing nearest neighbors. So in particular, what each of these black dots correspond to is a function that is comparing the test image with the training image. And this is done by embedding both of them and then taking a dot product to get the similarity. And then each of these is the label corresponding to each of these training images. And then what we'll do is when we're actually predicting the label for this test example, we're going to look at the similarity to each of the training examples and then multiply that with the label to predict the test image. And then what we can do is we can actually differentiate through this entire process, through this entire kind of nearest neighbors process to optimize this metric so that it accurately allows you to classify the test example as the correct class. Okay, and so some of the details here is that this is kind of a convolutional encoder that's taking as input the image and predicting this embedding. They also use a bidirectional LSTM here in order to kind of share information across the images when learning this embedding. And this whole thing is trained end to end, and it's trained such that when you do nearest neighbors in this embedding space, it actually gives you the correct answer when you try to do this n-way one-shot classification. Okay, and so here meta training and meta test time actually match because this is exactly the procedure that you'll then do at meta test time when you're given a training set and a test set, is you'll run it through this network and do nearest neighbors to get the correct answer. rather than treating meta training as a binary classification problem. Okay, so what does this look like as an algorithm? It's going to look very similar to optimization-based meta-learning, where we sample a task and sample a train set and a test set for that task. However, what's going to be different is instead of fine-tuning the model on our training data set, we're instead going to use this nearest neighbor predictor using our model F. And the parameters phi I, the task-specific parameters, are kind of integrated out here. And hence, this is why it's called non-parametric, because we're not actually explicitly representing phi I anywhere in here. However, we are still using this parametric meta-learner, which is represented by F theta. Okay, so then we use this to compute the predicted label for the test examples, and then we can update the parameters of our meta-learner such that the predicted labels and the correct labels match. Okay, so this is kind of the basics of matching networks and this sort of non-parametric meta-learning approach, where we are doing nearest neighbors in the space and then training the parameters of this metric space so that nearest neighbors in that space works well. Now, what if you have more than one example per class? So one challenge that comes up is in matching networks, we're doing this comparison to each of our examples. And if we have more than one example per class, we're essentially just going to independently compare X test to each of the examples in that class. And so one thing that would be nice to do is instead of performing these comparisons independently, can we aggregate information across the class so that if, for example, it's not actually directly similar to any one of the training examples, but it's actually similar to them in aggregate, then we'll still get the correct answer. And so what we're going to try to do is aggregate class information across each of the examples in our task training set to create a prototypical embedding of that class. And then we're going to compare our test example to that prototypical embedding. And so this is the idea behind prototypical networks. And in particular, what we're going to do is we're going to take the training data set for a task, which corresponds to each of these colored circles. We're going to, for one class, we're going to compute this prototypical embedding of that class, and we're going to do that for each of the n classes. And then once we compute this embedding, we're then going to just do nearest neighbors between our test example and each of the prototypes, rather than doing nearest neighbors between our test example and each of the individual examples. And this is important because if you look at this image right here, and this is your test example, if you just did independent comparisons, you might actually think that class 3 is correct because you're slightly closer to this example than to this example. However, once you actually aggregate this information and compare it to prototypes, you'll actually realize that the correct answer is the second class right here. And if you actually write out what this kind of nearest neighbors to prototypes looks like, it essentially will correspond to this equation right here. Okay. So really the key idea behind these non-parametric meta-learning methods is to essentially embed all of your examples and then do some form of nearest neighbors to the examples in your training data set, either with a assigned network, with this sort of matching network, or by embedding the examples in the prototypes and then doing nearest neighbors to the prototypes. Okay, so how does optimization-based meta-learning compare to non-parametric methods? One perspective that I think is useful is what I call the computation graph perspective, which is that you can actually write out these meta-learning methods as some overall function that takes as input the training examples and the test input, and does some sort of optimization on the training example, such as fine-tuning, and uses that to predict for the test example. And then you backpropagate essentially through that computation graph back to the parameters data. And so in both kind of optimization-based meta-learning and non-parametric meta-learning, you can view both of them as essentially some computation graph that takes as input the training set and the test set, and backpropagates back to theta. It's just a different computation graph for the two of them, where one computation graph involves gradient descent and another computation graph involves some sort of nearest neighbors. And so from this perspective, these methods look potentially a lot more similar, but they're essentially just embedding different sorts of structure into the computation graph to encourage it to do different things in different scenarios. Okay, so now to summarize kind of the pros and cons of both of these optimization-based methods as well as non-parametric meta-learning methods. For optimization-based methods, we saw that it has this kind of positive inductive bias at the start of meta-learning because it just reduces to gradient descent. In practice, it also handles varying and large numbers of shots well. And it's also model agnostic. It's also pretty expressive for deep models. And the downsides are that we have the second order optimization and it's usually fairly compute and memory intensive. Whereas for non-parametric approaches, they're also quite expressive. And one of the big benefits is that they're entirely feed forward. You don't need this sort of second order optimization. You just have a feed forward process that embeds everything and then does this comparison in that embedding space. And this means that it ends up being a lot cheaper and faster to optimize these sorts of models. And it doesn't require this bi-level optimization. Under certain conditions, it will be what I call consistent, meaning that as you get more training data for your test task, it will eventually get the correct answer. And then the downside is that it's harder to generalize to varying numbers of shots, varying K, and also hard to scale to very large K because non-parametric methods are very expensive because you have to do all these sorts of comparisons. And then so far, these methods have been limited to classification because you're doing nearest neighbors to different, and then predicting the corresponding class label. And so, for example, if you have a regression problem, then you can't do nearest neighbors for regression problems. Okay, and then generally well-tuned versions of both of these meta-learning methods can perform comparably on few shot learning benchmarks. This likely says more about the benchmarks than the methods themselves, and which method you use will probably depend on your use case and whether things like computational efficiency is really important or whether you have a regression problem and, for example, optimization-based meta-learning would make a lot more sense. Okay, cool. So I went through that relatively quickly. Any questions on non-parametric meta-learning methods or on this comparison between methods before I move on to applications?
So there is one question. Are these networks sensitive to shifts, rotations, or other noise?
Yeah, that's a good question. So it really depends on the data. So if the data has shifts, rotations, and other noise and so forth, then it can learn to be robust to those things. If it doesn't have that sort of thing, and then you test it on data that is noisy or has slightly rotated and so forth, it can be quite sensitive to each of those things. One idea is if you use things like data augmentation and artificially add random shifts and random rotations and so forth, that can help these models be a lot more robust. Yeah.
Sounds great. So there is another question now. So can you repeat where the second order optimization comes in?
Yeah, so.
And is this, sorry, there is just one more from the same, from Henry as well. So, and is this straightforward to implement?
Yeah, so I guess first with regard to implementation, both of these methods are, I think, relatively straightforward to implement using tools like TensorFlow and PyTorch. I think that like the first time that I implemented an optimization-based MetaLearner, it took me like less than a day to implement and run on a simple problem. And there's also existing implementations as well. And there's actually also libraries, like there's a library called Higher in PyTorch that is pretty useful for things like second order optimization and higher order optimization. So yeah, there's open source implementations. It's also not too hard to implement using these deep learning libraries. If you don't use something like PyTorch and TensorFlow, I think it's a lot more difficult to optimize because you need to compute all the gradients and so forth. But those libraries are super, super useful and also pretty fast. And then with regard to the question of where the second order optimization comes in, to be clear, this doesn't come in with approaches like matching networks and so forth. These are a first order optimization. It only comes in optimization-based metalearning methods. And the place where it comes in is that we, when we have this objective right here, you'll note that we typically optimize this full objective with gradient descent with respect to theta. And so when we run gradient descent on this, we're going to have a gradient with respect to theta. And we also have a gradient with respect to theta here. And when you multiply the two together, you get a second order gradient term.
Thank you. Yeah, we're good to go.
Okay, great. And we'll also have time for questions at the end of the coffee break as well. Okay, so now we'll talk about some applications and how this has actually been used, how these methods have actually been used in practice in biological applications. So I'll talk about both metalearning for drug discovery problems, as well as cellular response prediction. And the goals here will be to help kind of figure out how to apply metalearning to your own problem and familiarity with some of the applications in biomedicine. And so far, we've really only talked about these image recognition examples, and a lot of approaches are only evaluated on image recognition benchmarks. And so can we apply these methods beyond computer vision problems? So first, we'll typically need a different backbone network. A lot of image classification problems use convolutional networks. And so instead, we'll need things like graph neural networks or LSTMs and so forth. And this backbone neural network is going to depend on your application and the structure of your data. So how do you get started? So there's really kind of three key steps. The first is to define your tasks. And the more tasks you have, the better. And the tasks should share some structure. Then you should choose the method that you're going to try. If you have a classification task, then you can use either optimization-based or non-parametric. If you have a data that's kind of more compute intensive, then you may want to choose non-parametric. Whereas if you think that your tasks might be a little bit more out of distribution, it might be better to choose optimization-based methods. Whereas if you have a regression problem, you should choose an optimization-based metalearner. And in general, it's a little bit hard to know in advance, and it's usually somewhat problem dependent. So you may also want to just try both approaches as well. And then lastly, just to choose a backbone network architecture, and this is going to depend on your data type. If you have a graph, then graph neural networks are a great choice. If you have sequences, then things like LSTMs and recurrent neural networks can be useful, and so forth. Okay, so let's first look at this application in drug discovery, and we're going to particularly look at this paper from Altetron et al. In 2017. Although there's also another paper that we're not going to cover, and if you're interested, you can take a look at it. And so the problem here is we want to be able to predict the activity of a molecule in a new experimental system or assay. And the challenge here is if we have really a small amount of biological data available to get accurate predictions for these new compounds, then we want to be able to generalize with such a small amount of data. And so kind of the discovery of new drugs corresponds to this low data problem. However, we do have a fair amount of data from other molecules and other assays and so forth. So the goal here is, given the molecular structure of the compound, we want to be able to predict the experimental outcomes for different assays. And different tasks are going to correspond to different molecules and different experimental assays. And so the goal here is we want to be able to generalize across these different assays, and in particular generalize to new assays with only a little bit of data. So why is this hard? In object recognition, we're just trying to generalize to new classes of images, whereas in this case, we're actually going to try to be generalizing to related but not identical tasks. and non-identical assays in our training collection. And so this really requires generalization to completely new tasks, which are kind of completely different classification problems, essentially. And so what this paper did is it extended matching networks to this task. And so matching networks, this is the second non-parametric meta-learning approach that we saw. And they kind of course looked at a data set with different compounds. They had labels for different assays for that compound. And they encode the structure of the molecules of the compounds into some embedding space. And then use this context-based attention LSTM architecture to process this data into and update the embeddings, and then compute some similarity function between the new compound and the previous compounds. And based on the similarity between this embedding of the new compound and the embeddings of the existing compounds, we compute these similarity scores and then take the corresponding labels. And for the compound that we're most similar to, we output the corresponding label, which is this kind of red label right here. And then, of course, this is what happens when we're trying to perform this few-shot classification from this assay. And then during the meta training process, we take all of our existing data and essentially backpropagate through the encoder, through the LSTM, and so forth to optimize all of the parameters of this process so that we accurately predict the property for the new compound. Okay, so how do we actually go about encoding these molecules? The idea here is to use a form of graph neural networks to transform these small molecules into vector representations. And once we have these vector representations, it's easier to use neural networks to process them. So we take as input our molecule, extract the graph topology and the atom features of this molecule. And then we can once we have this graph, we can apply graph convolutions and pooling to the graph. And then apply this graph gather operation, which will essentially just take all of the nodes of your graph and sum the embeddings to form this representation of the graph. And then once we have this vector representation, we can apply dense fully connected layers to get more complex representations and more process representations of the graph or of the molecule. Okay, so this is kind of this molecular encoding function. And kind of to summarize the approach again, once we have this molecular encoder, we apply a context LCM and the similarity function to actually predict the property of the molecule with these kind of small amounts of data. Okay, so in their experiments, they looked at 3 different data sets. They looked at these nuclear receptor assays that are related to toxicity, and they meta-trained on 9 different assays and then evaluated on 3 new assays. They also looked at the CIDR data set where that corresponds to different marketed medicines and their observed adverse reactions. And so in this case, they had 21 different organ classes, and then they evaluated on 6 new organ classes. And then finally, the last data set looked at assays used for standard virtual screening. And the positive examples were selected to be kind of structurally distinct from one another. And one thing that you'll note here is this is still a relatively small number of meta training tasks, so 9 or 21 or 17. And as we'll see in the results, they're actually able to effectively leverage this prior data in a way that allows the model to make correct predictions at meta test time. Okay, so during training, they construct episodes that try to mimic this test setting, just like we talked about before, where you randomly sample an assay from the training assays, sample a train set and a test set. So the support set or the training set consists of kind of a positive and a negative example from the selected assay. And then they also sample this test set, and this is what the loss function will be computed on. And then at evaluation time, they also randomly sampled the support set from each test assay and then evaluated the performance on the remaining data points. And they kind of repeat this process for different random selections of the support set to evaluate how well it can handle different support sets and different training sets for each assay. Okay, so they compared metalearning to standard supervised machine learning, which is just a single task approach that considers a single assay, which is just trained on these two examples, one the positive and negative example, and evaluate the remaining data points. And they tried both a random forest as well as a graph convolutional network. And then they also compared their matching networks approach to a Siamese network that is trained in it and evaluated in a similar fashion to the matching network, except using this different Fusha learning approach. Okay, and so what we can see, the results here is first, on both the Tox21 and CIDR data sets, we see kind of a pretty significant performance boost using this few-shot matching network approach compared to both supervised learning approaches as well as the Siamese network approach. And so this means that first, we see a pretty significant benefit from using the data from other assays. And second, we see that with this comparison to Siamese networks, we see that by actually making meta training and meta test time match, we can do significantly better than if we were to just construct these binary classification problems during meta training. And then on the MEV data set, we see that these simple baselines are better. And this might be because it contains these really structurally distinct assays, indicating that the methods are really struggling to generalize in this setting. And if you are kind of interested in kind of the more details and more comprehensive evaluation, you can take a look at the paper. Okay, and then the second application that we'll look at is the cellular response prediction task. And so the motivation here is in drug development, you have typically these different stages, starting with in vitro cell cultures to more advanced culture systems and animal models, and then finally to human cohorts. And of course, in the in vitro cell cultures, it's really easy to get a lot of data. It's through high throughput screening and so forth. And as you kind of go further into the process, it gets much harder to get labeled data. And so what we'd like to be able to do is we'd like to be able to essentially translate biomarkers from basic research to clinical utility. And this is going to involve kind of transfer of information across a series of contexts. So how do we transfer the drug responsive cell lines across these different contexts? So we can frame this as a few-shot learning problem. And the key idea will be to try to learn to generalize using abundant human cell line data and use that to try to generalize with a small amount of examples to kind of the later contexts. So they looked at 3 different drug response transfer problems. The first was transferring across different tissue types. The second was to transfer from tumor cell lines to patient-derived tumor cell cultures in vitro. And the third one is to transfer from tumor cell lines to patient-derived tumor xenografts in mice in vivo. And so essentially, as you go along, this transfer problem is going to get harder and harder because you're kind of moving towards things that are kind of more different from what you saw during meta training. Okay, so in the metalearning stage, where the different tasks they correspond to was different tissues and trying to learn how to generalize across these different tissues with small amounts of data. And essentially the features corresponded to features of different cell lines, and then the labels correspond to the drug response prediction. And yeah, like I mentioned, that kind of the metadata set or the set of tasks that they're corresponding to is different cell lines across different tissues. We have different molecular features, and then we're looking at tumor growth. In the paper, they also considered a gene deletion response task. And if you're interested in that task, you can also take a look at the paper. And so, yeah, the goal is to adopt to new tasks from a few labeled cell lines in a new tissue or in a new context. And so, like I mentioned before, there's these 3 transfer scenarios. So once we do the meta training process, we'll either want to transfer to a new tissue, transfer to a PDTC model, or transfer to a PDX model. So the metalearning method that they used here is an optimization-based metalearner, or the MAML or model-agnostic metalearning method that we talked about before. The backbone architecture that they use is a fully connected neural network, and the label corresponds to predicting tumor growth. So this is a regression task. And so this means that it makes sense to use an algorithm like MAML because things like prototypical networks, matching networks won't be applicable. And then the loss function corresponds to mean squared error. And then once they perform this meta training process and adapt it to the different scenarios, they looked at the Pearson correlation between the predicted response and the actual drug response as their evaluation metric. And they trained a separate model for each drug. Okay, so what does our experimental setup look like? They randomly sampled a subset of tissues and sampled a support set consisting of 6 cell lines, and then a query set of new cell lines that the loss will be computed on. And then at evaluation time, they randomly sample K cell lines as the support set from a held out experiment in a new context, and then they evaluate the remaining cell lines that exist. And then they repeat this for different random selections of the support set. Okay, so they compared their the metalearning approach, the MAML approach, to different kinds of models, different standard supervised learning models that are just trained on the support set. The test example, and in particular, they trained a random forest, a neural network. They also tried nearest neighbors and linear regression. And one of the things that they did is actually, instead of actually just training on the cell lines from the target task, they actually combined the data from all the training tasks and evaluated on the test data in the new biological context. Yeah. Okay, so what these plots are showing is the metalearning approach is in red. It's called TCRP. And then each of the supervised learning approaches, random forest, neural network, nearest neighbor, and linear regression, are shown in these like blue and green and purple colors. And what we see is as we kind of increase the number of samples from 0 to 10, we kind of see an increase in the correlation. And overall, we see kind of a pretty substantial difference between this metalearning method and methods trained with supervised learning. We also see some performance variation across different drugs. And this is kind of as we would expect, since different drugs will probably share different amounts of structure and be easier or harder. Okay, so to summarize, we talked about optimization-based metalearning and non-parametric metalearning, and we also looked at a couple different applications. If you're interested in learning more, here are some of the really key references that we covered, including model-agnostic metalearning, including these non-parametric metalearning methods, as well as these two application papers. And up next, we'll be talking about prior knowledge enhanced models. But first, we can go through some questions before the break.
So there is one question regarding the drug discovery problem. Does the module encoder stays the same for the test data?
Yeah, the encoder does stay the same. That may have been a typo. Yeah, so I believe the encoder stays the same. I think that one reason why it may be, right, so the reason why these are labeled as slightly differently is that this encoder actually is is not just encoding each of these compounds independently, but it's actually encoding all of them and actually leveraging information across these structures, across these compounds, whereas this encoder is just encoding a single compound. And so these are actually different encoders, but between meta training and meta testing, you're going to be applying G prime to the support data that you see at meta test time, and F prime to the unlabeled test examples that you see at test time.
Great, thank you. I think it seems there are no more questions. So any other questions before the break? Why do you want to connect different drugs in G prime? Right,
so the reason why this can be helpful is that essentially when you're classified between two things, you could just kind of encode things independently, but depending on the kind of task you have, it may be helpful to actually look at them kind of collectively, because, for example, I can maybe give an example in image classification where, say, your two examples, maybe you have a dog and a cat, you're trying to classify between dogs and cats, then you may actually encode things differently, encode things in one way in that setting. But if you're trying to, for example, classify between two breeds of dogs, then you want your features in that more fine-grained classification problem to represent those features more finely. And so depending on how fine-grained your classification problem is, actually looking at the examples can be helpful to figure out what your embedding should look like, as opposed to just looking at them independently.
Hi, welcome to the second part of the tutorial. And in the next part of the tutorial, we will explain how can we incorporate prior knowledge into Fusha learning methods. So topic-wise, we are still on the Fusha learning methods, but now our aim is to kind of extend these methods by incorporating prior knowledge. And we will consider two levels of prior knowledge, task level and feature level, that we will explain in the next slides. So in the summary, in the next part of the tutorial, we will explain how can we incorporate prior knowledge into Fusha learning methods. So we will discuss feature level and task level prior knowledge, and we will show how can we build interpretable models by incorporating feature level prior knowledge. And after introducing the methodology, we will consider applications of these methods for annotating cell types in single cell data sets and for disease predictions. So the learning goals of this part of the tutorial are to first understand the types of prior knowledge and where to get the data. And the good news is that this data is usually readily available in biomedical applications. And our second learning goal is to understand prior knowledge enhanced methods, how to implement them, and how you can apply them to your own problem. So far we talked about the concept of meta-learning and general Fusha learning methods. And we covered optimization-based methods such as MAML and non-parametric methods such as Siamese networks, matching networks, and prototypical networks. But what if we have additional background knowledge about the classes or features, such as, for example, metadata or relationships between the features or classes? Can we somehow use this information and build better models using that information? And by better, I mean models that can kind of further improve performance on Fusha tasks, and ideally the methods that are not just the black box models. So the key idea we will present next is to use prior knowledge to enhance existing Fusha learning methods we covered in the first part of the tutorial. But first, the question is kind of where to get this prior knowledge. And the great news is that in biomedical applications, this knowledge is very often readily available. So there are many ontologies and knowledge bases built with a huge effort of the community. For example, a disease ontology has been developed as a standardized ontology for human diseases with the purpose of kind of providing the biomedical community with consistent descriptions of human disease terms. And in the example shown on the slides, if we kind of check the heart disease, for example, we can see based on the disease ontology that heart disease is a cardiovascular system disease, and it is also complex genetic disease. And if we go further down this branch, we can see that the heart disease is also hereditary disease. And then based on the cell ontology, we can look at another branch, which is kind of congenital chromosomal disease. And we can see that these two diseases are both hereditary diseases. So kind of the idea here is that we can use these relationships between diseases in this case to improve our predictions, in particular when faced kind of with rare diseases for which we cannot gather enough of labeled examples when we don't have sufficient information. Another example is kind of cell ontology, which is designed as a structured control vocabulary for cell types and defines relationships between the cell types. Or for example, gene ontology, which defines relationships between gene functional terms, and kind of we can map the genes to gene ontology terms and obtain information about functional similarities of genes. So these are just few examples of ontologies, but there are many other knowledge bases in biology that we can kind of reuse in this case. And the prior knowledge can be given and incorporating into the model on the following two levels: task level and the feature level. So the idea in the task level prior knowledge is that we have kind of information about class attributes or relationships between the classes. So in the machine learning sense, kind of tasks are defined as classes we are trying to predict. For example, which disease does the patient suffer from? And following that example, we might, for example, use information that both coronary heart disease and stroke are cardiovascular diseases, or we might have descriptions of each class, such as text for a scientific article or Wikipedia article which describes particular disease. And by feature level prior knowledge, we consider the setting in which we have prior information about relationships of features. So in disease prediction problem, features might be genes, and we could kind of use, for example, gene ontology to define feature level prior knowledge. So let's illustrate now feature and task level prior knowledge on the cell type annotation problem. So in this problem, our learning examples will be cells and our features will be genes. And our prediction task is given gene expression of cells. We want to kind of predict the correct cell types. And since genes are our features, we can kind of consider to incorporate gene ontology as our feature level prior knowledge and use it to define functional similarities between the genes. Or for example, we could incorporate protein structural similarities from PDB or SCOPE database. And what can we use as the task level prior knowledge in this example? So our task is, in this case, is to predict the cell types. So we could kind of use the relationships between the cell types from the cell ontology. Or for example, if we want to describe each cell type, instead of modeling the relationships, we might incorporate some metadata about task attributes. In this case, these might correspond to kind of marker genes of cell types. And now that we're kind of equipped with the concepts of task and feature level prior knowledge, we will next discuss how can we incorporate this background knowledge into few-shot learning methods. Are there any questions so far? Okay, we are good to go. So we will first start with the method for feature level prior knowledge. So let's first do a recap of prototypical networks covered in the first part of the tutorial, because we will build all our methods in this part of the tutorial on top of the prototypical networks. And the main idea in prototypical networks is to kind of learn an embedding in which points cluster around the single prototype representation for each class. So given, for example, an image as an input, we will learn nonlinear embedding function f using deep neural networks that will map this image into low-dimensional space. And we want to achieve that the image is mapped close to the prototype or centroid of the class it belongs to. So in the example on the slide, we have 3 class prototypes, which are visualized here as these green, red, and blue circles. And in this toy example, this image belongs to this blue class. So kind of we want to embed it close to this blue centroid. And mathematically, we can simply compute the prototype for each class just as the mean embedding of the support set, where support set kind of consists of the few labeled examples introduced in the first part of the tutorial. And given class prototype, we can then compute the probability that the query data point, denoted here as this XQ, belongs to a particular class by looking at the distance between kind of class prototype and embedding of the query data point. And we will normalize this by across all other classes to get the probability function. And note that the distances here are always computed in the embedding space because these prototypes are found in the embedding space, and we embed our query data point using f theta. And for the distance function, we can really use any distance function, but the most often used is either a Euclidean or a cosine distance. So to recap the prototypical algorithm, the idea kind of is that the training will proceed in episodes, and we want to formulate an episodes in a way that it resembles what we will see during the testing. So each episode is formed kind of first by sampling the task, meaning sampling classes from the training set that define our task or prediction problem. And next, we sample data points from these classes, and we divide them into two disjoint data sets: support set, which consists of a few labeled examples, and the query set, which consists of data points whose labels are used to compute the prediction error. And kind of given the sampled support set, we will first compute the prototype as the mean embedding of the support set. And we will then minimize the loss on the sampled query set in each episode. And the key idea behind this meta-learning training scheme is to kind of improve the generalization of the model by trying to mimic the low data regime, which we will encounter during tests. And next, we will extend this approach by incorporating feature-level prior knowledge. And the key idea in the concept-based meta-learning approach, named COMET, is to learn embeddings along high-level human interpretable concept dimensions. So instead of learning a single prototype for each class, what we want to do is we want to learn a set of prototypes, one for each concept dimension. So following a bird example from previous slide, we first start by identifying part-based representations of the input that are defined as human interpretable concepts. So in this case, this concept might correspond, for example, to bird's beak, bird's wing, or bird's leg. And then instead of learning to embed whole image in the embedding space, we will embed each part into its concept embedding space. And we will call these embedding functions concept learners. And kind of finally, each class will be represented instead of with the one single prototype, as in the prototypical network, it will be represented with a set of concept prototypes. So our image in this example will be embedded into three concept spaces, so each denoted in this circle, and we want to kind of embed it in a way that data points cluster around correct concept prototype in each concept space. And in summary, the main idea here is to learn a unique metric space for each concept dimension, and then somehow combine these independent concept learners. And we will explain in the following slide how can we combine these concept learners. But before that, the question is kind of what do these concepts really correspond to, and how can we obtain them? So we can see that this concept as subsets of related dimensions or features. So in image classification that we've seen in the example, concept might be part-based representation of the input, such as, for example, bird's beak, bird's wing, or leg. And if genes are R features, we might kind of group genes using existing knowledge bases based on functional or structural similarity. If cells are R features, maybe we can group them based on the cell type similarity. So how to obtain the concepts? The concepts can be obtained from existing knowledge bases, for example, gene ontology or scope, if we want to use gene as features. And if we can't get this concept from the background knowledge, it is also possible to kind of use unsupervised learning approaches and to discover concepts. And let's now see how can we compute the concept prototypes. So mathematically, we will define concept as a binary 0-1 vector, and it will have the same dimensionality as our input. And the value in the vector will be 1 if we want to incorporate a particular dimension in the concept, and we will have 0 otherwise. For example, if our concept is a particular gene ontology term, then all the genes that belong to that geo term will have value 1 and 0 otherwise. And the prototype of concept J is then computed as the mean of concept embeddings of the support example, where support example is transformed based on the concept J. And the transformation is simply defined as the element-wise product between example and the concept. So recall that the concept is 0-1 vector. So this means what we will do is we will zero out all dimensions in X that do not belong to a particular concept. And given a concept J, we can then compute a prototype for each class. And finally, we have all these concept prototypes. And the next question is how we can combine these prototypes to get the final prediction. So given a query point, we will first estimate distances to each of the concept prototype similarly as we did in standard prototypical networks. But just in this case, instead of single distance, we will get one distance for each concept space. And here, this query data point is shown in this as a gray circle because we don't know its class. So given the embedded data point, we will look how far is it from each of the concept prototypes. And we will repeat this approach for each concept. And we will next combine these prototypes by taking sum over all concept prototypes and normalize it across all the classes. So the inner part here looks at the distance between concept embedding of the query data point and the concept prototype, and then we sum over all the possible concept prototypes and normalize over the classes K prime. And can we use this concept to go beyond the prediction tasks and get interpretations behind our predictions? And interpretability is, when you think about it, is particularly important in the Fusha setting because our predictions are really based only on the few labeled examples. And we will take the following approach to explain our prediction. So for each concept, we will estimate concept importance scores based on the average distance between concept prototype and the concept embedding. So intuitively, lower the distance between concept embedding and concept prototype, we will have higher importance score. And higher score indicates higher contribution in classifying that data point to a particular class. For example, let's say we have 3 concepts and 3 classes shown again in the green, red, and blue color. And we assign the test data point to the blue class. So what we will do is at each concept space, we will look the distance between the concept embedding of the data point and its concept prototype. And we use distances to define importance scores and rank them. So in this case, the data point which has the smallest distance to the concept prototype is in the concept 1 space. So this concept will get the highest importance. And the second lowest distance comes from this concept 3 space. And so we will rank it as the second most important concept. And finally, the concept 2 has the distance is the largest between the concept prototype and the data point in this concept space. So we will rank it as the third, the least important concept. And we will next show how can we apply this approach for cell type annotations in single cell experiments. Are there any questions at this part?
Maria, there are 2 questions on learning the concepts. So Eric asked how the concepts are learned automatically, and Marcin asked if the concept learners share parameters.
These are both great questions. So If you want to kind of do unsupervised discovery, so this also again depends what type of data you have. So if you have the images, there are some approaches, also you can check the paper, and there are some approaches which kind of do unsupervised concept discovery in case of the images, where you try to kind of find the landmarks on it across different images. If you have kind of genes as features, you can maybe try to kind of cluster the data, find some structure before in the data, and then give it as an input to the algorithm.
There's one other question by Shaman who asks, can the concepts overlap or intersect with each other?
Yes, they can, and we will see this actually in the experiments. So yes, the concept can be kind of noisy, they can be redundant, and the idea is that the algorithm automatically learns which of these concepts are important for the prediction. And regarding the second question we have is, yes, the network can share the parameters, and there in the paper, there is also like experiment between if you share the parameters and if you have the different parameters across concepts, and you get slightly better performance if you have kind of different parameters, but still the performance is very high even if you share the parameters. Okay, are we good to go?
Yeah, I think there's one other quick question on, are the prototype concepts similar to cluster centers?
Yes, you can see them as the cluster centers.
That's all.
Okay, great, thank you, Yusuf. So let's see how can we apply this approach for cell type annotation problem. So let's first do a very brief introduction in the single cell genomics for those of you who are not familiar with it. So with the advances of single cell sequencing technologies, we can kind of sequence the genome or transcriptome of the individual cell and study mutations carried by the small number of cells. And compared to traditional sequencing technologies that sequence and take average expressions of population of cells, single cell technologies allow us to kind of study the heterogeneity on the resolution of individual single cells. And this heterogeneity of individual cells is visible, is not visible if we kind of look at the population of the cells, but it's only visible if we go on the single cell level. So the standard approach is kind of do the tissue dissection, isolate the individual cells, and finally we get the cell by genes matrix, and each cell is represented with a very high dimensional gene expression vector. And this matrix is usually very sparse, meaning that most genes will have 0 values assigned. And the central fundamental task in analysis of the single cell data is the identification of cell types. So this means that we want to kind of automatically classify cells into different groups, such as B cells, T cells, and fibroblasts, for example, based on their gene expression profiles. So as an input, we are given a matrix, which is cells times genes, and we want to kind of label each cell according to its cell type as shown here. So in the machine learning sense, simply cells are learning examples, genes are features, and cell types are our labels. And there are a number of challenges which arise when you deal with the single cell data sets. So first, there are many poorly identified cell types and cell states, meaning that we only might have few labeled examples that belong to a particular class. So secondly, there are many rare and unseen cell types. And the reason is that our knowledge is still incomplete in this space, and some of these cell types we cannot confidently label. And last but not least, we are faced often with kind of heterogeneous experimental conditions. For example, we might do the sequencing using one sequencing technology and then get another data set using another sequencing technology. And then we can see the clustering based on the technology and not based on the cell type. And it's shown here in the example. So red color shows one technology, while cyan color shows different technology. So although these data sets are coming from the same tissue, we see kind of the shift only because using different technologies. And these problems are even more amplified and even more challenging if we want to kind of compare different species or different diseases. And given only few labeled cells per cell type, the question is whether performance on this kind of cell type annotation task can be improved if we use gene ontology as prior knowledge. And gene ontology groups genes based on the functional similarity. So we could kind of use GO to define concepts and group genes into functionally related groups. And kind of if genes is assigned to a particular GO term in terms of the earlier introduced comment, it would get a value 1 in our concept vector and otherwise 0. So kind of the key idea here is to exploit this gene ontology by grouping functionally similar genes and then apply comment for the few-shot learning task and see whether we can get improvement compared to other few-shot learning baselines. So the dataset considered in the paper is the Tabula Muris mouse cell atlas, which contains more than 100,000 cells, which are sequenced from more than 20 mouse tissues. And in total, there are more than 100 different cell types across all the tissues. And the task that we are trying to solve is to kind of generalize across different tissues. So given a new tissue and then only few labeled examples per cell type, the method needs to learn to generalize to new cell types across tissues. And in COMET, concepts are defined, as we said, using gene ontology, and the backbone network used is a fully connected neural network, which consists of the 2 layers, and kind of the same backbones is applied to the COMET and other baselines. So experimental setup is similar to applications we considered in the first part of the tutorial, but let's do the recap. So kind of the idea during training is to construct the episodes so that our training process mimics what happens during test. So first, cell types will be randomly sampled to define a task in each episode, and then given a task, disjoint support and query sets are computed. And in case of one-shot learning, support set consists of 1 positive and 1 negative example per class. And then query set is used to compute the loss and update the parameters. And during model evaluation or testing, the model is evaluated on the so-called 5-way setting. So 5-way here means that at each iteration, we sample randomly 5 cell types. And if we are solving the few-shot learning task, then the support set will have the 1 positive and 1 negative example. And then the performance is evaluated on the query set, which comes from the same set of cell types as the support set. So in summary, kind of given 2 labeled data points, 1 positive, 1 negative, the model needs to generalize to these data points in the query set. And the whole procedure is repeated to obtain mean performance across different random selections. So the following baselines are considered to compare performance between prior knowledge enhanced method COMET and state-of-the-art methods. So fine-tune baseline means kind of that we will pre-train the network using all available labeled data and then just fine-tune on the few labeled examples on the new task. Matching networks and prototypical networks are non-parametric methods that we covered in the tutorial. Relation networks is kind of the newer metric-based approach that kind of learns relation scores between the query examples and the support set, while MetaOptNet is one of the strongest baselines that learns feature embeddings under a linear classification rule for novel categories. So they are out of the scope of this tutorial, but we provide all references at the end of the tutorial so you can kind of explore these methods in more details if interested. And finally, ProtoNet ensemble is kind of constructed by building ensemble of prototypical networks. Kind of since COMET can be seen as an ensemble-based approach, the idea is to compare performance to kind of just building an ensemble of prototypical networks. And ensemble here means that we will train a number of prototypical networks, and then we can use the voting scheme to decide on the final prediction. And the results show that we can get significantly better performance in this task if we use a prior knowledge enhanced method COMET in both 1-shot and 5-shot setting. And in the 5-shot setting, accuracy we can see is over 90%, while in the 1-shot setting, it is almost 80%, which is 6% improvement over other second-best method. And when looking at the performance as the function of the number of concepts, where 1 concept means kind of standard prototypical network, the performance consistently improves with more concepts. And kind of since gene ontology is a tree and related to earlier question, kind of these concepts will be highly redundant because they will be selected basically at different, by cutting the tree at different levels. So that means that kind of with more with 1,500 concepts, we will cover large part of the geo tree. And the results show that even with more concepts, we kind of, even though these concepts are highly redundant, we are still able to get somewhat better performance, especially on the Fusha task. And how to get explanations behind the predictions, and do these interpretations make sense? So since gene ontology is used to define the concepts, Comet will rank these gene ontology terms by assigning concept importance scores to each gene ontology term. And we need to somehow evaluate whether these gene ontology ranking is meaningful. So we need to obtain the ground truth terms. So the way to do that is kind of, for each cell type, we can find the genes that are differentially expressed in that particular cell type. And then we can run the gene ontology and terms enrichment and see which gene ontology terms are enriched in the ground truth cell types. And then we can kind of compare the Comet's range. Once we obtain kind of the set of genes, once we obtain the set of gene ontology terms, we can compare these rankings based on the Comet's predictions. And the results show that Comet kind of reconstructs well ground truth terms. So these ground truth terms are shown here in the pink color, while the Comet predictions are shown in the blue color. So, and the Comet is able to achieve 0.7 recall at the top 20 highest terms. So next we will explain how can we incorporate prior knowledge on the task level into existing methods. So any questions on the earlier part?
Yeah, Henry had two questions. His first question is, are concepts functionally related genes? And can genes be grouped exclusively? Meaning, can you have a common gene in like two concepts?
Yes, you can have a common gene to concept. That will be the case if you use gene ontology. And the answer to the first question is again, yes. So these genes will be kind of functionally, the concepts will be based on the functionally related terms. But if you go kind of higher in the hierarchy, this you will kind of get more coarse or labeled relations between the genes.
Yeah, and his second question, I think this might be a typo. In slide 24, it has 1 plus twice in the evaluation section.
Slide. Yes. So here should be 1 positive and 1 negative example. Thank you.
That's it.
Okay, thanks, Yusuf. Okay, let's see next, how can we incorporate prior knowledge on the task level compared to feature level, what we did earlier. And for simplicity, we will again consider methods that will be built upon prototypical networks. So we will first consider the setting in which prior knowledge is given as task level attributes. So let's use disease prediction example to explain what we mean by task level attributes. So let's say we want to predict diseases and we have information about the patients and their association to a particular disease. And then additionally, we have the task level attributes which take a form, for example, of the semantic attribute, meaning textual description of a particular disease. So for Alzheimer's disease, maybe we have description that this is a progressive brain disease that destroys memory, while Parkinson's disease is a brain disorder in the central nervous system that affects movement. So in this example, kind of by using textual description, the model can relate Alzheimer's and Parkinson's disease because both are brain disorders. Or for example, in the cell type annotation task, we might include marker genes as task level attributes. And this could kind of allow the model to regard cell types that share marker genes as the more similar to each other and reuse this information to build a better model. So the question is kind of how can we incorporate this additional class level information into our model? And the key idea in the adaptive cross-model mixture model, or AM3, is to kind of leverage prior knowledge by learning to combine distinctive embedding spaces. So there is one embedding space here, which is kind of generated using our learning examples by taking the mean of the support set, which is kind of done in the standard prototypical networks. But there is also other embedding space, which in this case is generated from the semantic information. And since these two embedding spaces have different structure, the proposed model kind of exploits both modalities adaptively. So the idea here is to augment prototypical networks by incorporating language structure. And this language structure here is captured by this embedding model W, pre-trained on unsupervised large text corpora. And word embedding model is then kind of followed by this function G. And this function G is needed to guarantee that the resulting semantic embedding will have the same dimension as this prototype. And we can kind of regard this semantic embedding as the prior knowledge prototype, and we want to combine it with this kind of prototype which we obtain from our support set. And the idea is to combine these prototypes adaptively by taking their convex combination with weights learned through adaptive mixing network. So mathematically, what we will do is we will first kind of take the convex combination of these examples. And then what we will do is we will learn this combination parameter lambda c through this adaptive mixing network age of vice c. And kind of finally, given the query data point, we will assign the probability by assigning the data point by looking at the distance between the query embedding and this kind of combined prototype instead of kind of only original prototype. Well, what would we do kind of if we can standard prototypical networks? So in the original paper, visual features are combined with semantic features, meaning kind of images with the text descriptions of the image. But extending this approach to different type of data modalities we might encounter in biomedical applications is not hard. So we could kind of be given any input and any class level attribute, but what we would need to change is kind of the backbone network, and instead of the convolutional and word embedding network, use kind of backbones that are applicable to our input type. And now when we understand kind of how to handle task level attributes, let's see what can we do when instead of the class level attributes, we have relations between the classes as the background knowledge. So domain knowledge might be given in the form of the graph that characterizes task relationships. So for example, if a model learns how to detect Alzheimer's disease, one can imagine that it is easier to modulate it to detect to kind of detect Parkinson's disease than some other less related disease. So the idea here is that the nodes which share the same parent are likely associated with the similar data points. And by exploring these kind of relations in our model, we could improve performance, hopefully, in the low data regime tasks. And while intuitively, when we think about it, it makes sense that these class relations might be useful in the few-shot learning, the question is, how can we effectively leverage and incorporate these relations into our model? So the key idea in the graph-enriched meta-learning approach proposed in the TADANET paper, which the approach is called TADANET, is to kind of to learn class prototypes that will enable message passing across classes based on the class hierarchy graph, which we define using external knowledge bases. So this method is again built upon prototypical networks. So first, we will kind of embed our data points and compute standard prototype by computing the mean of the support set. And we will then utilize the hierarchy graph to align classes in order to learn better class prototypes. So in the hierarchy graph, the target few-shot classes come from the leaf nodes, while the course nodes from the ancestors indicate the distance between the target classes. And the intuition here is that if two classes share the same parent node in the hierarchy graph, they are likely will have some common characteristics. So kind of their prototypes should be more similar. And class relatedness information from the hierarchy graph can be incorporated in the following way. So what we will do is that in each propagation iteration, we will update the prototype using the embeddings of the node neighbors in the previous iterations. So at the start, that is kind of at the iteration 0, we will first initialize the prototype based on the class embedding prototype. And then in each propagation iteration, what we will do is we will aggregate information from the node's neighborhood and then update the prototype. So the prototype is updated kind of by computing the attention score, which we can think of kind of as some similarity score between its own embedding from the previous iteration and the embedding of its neighbors. And kind of further modeling question is, how can we compute these attention scores? And also, how can we combine the prototypes? So the proposed way to compute attention scores in this paper is based on the distance between the neighbor embedding of the prototype and current class prototype. So in the paper, the distance function is the cosine distance, but you can use also other distance function. And the attention score we will again normalize over all neighbors of that class. So this means that the intuition is that closer the neighbor is to the target data point, it will have the higher influence. And finally, the idea is to kind of combine this embedding and propagation prototypes using weight lambda, which is a parameter set in the range of 0, 1. And weight kind of defines whether initial kind of standard prototypical embedding or propagation prototype should have higher influence. And by setting lambda to 0, we will get standard prototypical network formulation. And while kind of same as in all other methods, class probability we can again compute just by looking at the distance. Instead of kind of original prototype, we will look at the distance between this combined prototype. And the Tadanet approach we explained so far is based on extending prototypical networks by incorporating prior knowledge given in the form of graph. There are also other methods proposed for this kind of scenario in which you have graph information. One example is, for example, KGT and graph neural networks, which use graph convolutional neural networks to propagate class similarity information, while another approach is kind of based on incorporating graph regularization into objective function and proposing kind of model agnostic formulation that can be incorporated on top of any Fusha learning methods. And next, we will see how can we apply these methods for the problem of disease prediction and whether we can get better performance on this task if we exploit the relationships between the diseases. Any questions?
Yeah, there are actually 2 questions on the comment section.
Okay.
Oh, actually, just one just came in for the previous section. So let me take that first. So what kind of distance is used in attention mechanism?
So in this paper for Tadanet, it is the cosine distance originally.
Okay. Should we do the comment questions now? Or
sounds good. So,
okay. So the first one is from Akram, who asks, she doesn't understand how we can use Go terms for cell type annotation in single cell analysis.
So your features are, in this case, your features are genes. So the idea is kind of based on the gene expression profiles, you are predicting the cell types. So the idea is kind of that you exploit the information which genes are more similar to each other in order to improve in performance on these kind of cell type prediction tasks. So basically you are grouping genes into groups of related because your genes are kind of very sparse. You can exploit the information from the related groups to improve on this task.
And another question is, how are tissues used as tasks because they don't appear anywhere in the data matrix since you only have cells and genes?
So tissues are, so basically you will do the meta training over your tissues. So each tissue, in each tissue, you will have kind of different cell type annotation tasks because each tissue will have its own cell types. So you can kind of use the other tissues to learn to generalize to new tissue given few labeled examples.
And could you use cell types instead as tasks?
You can use cell types instead as tasks, yes. So we have to go forward. Do we have other questions?
I think they're just specific ones, so we can take that in the chat.
Yeah, I think in the interest of time, let's move forward and I can also answer those later. So next we will see how can we apply graph-enriched task level method Tadanet for predicting diseases from few labeled examples. So collecting large amounts of labeled patient data to train machine learning models to predict diseases is usually very hard, especially for rare diseases. And in this setting, kind of having machine learning methods that can generalize given small amounts of labeled training data is of crucial importance. And relations between different diseases are already given in kind of various databases such as disease ontology, and we can naturally ask whether using these relations can help us to achieve better performance on the disease prediction task when faced with a small number of labeled examples. And the dataset which is used in the Tadanet paper is the MIMIC3 dataset, which is kind of large, freely available dataset of patients admitted to the critical care units. It has over 40,000 patients collected during the period of 10 years. And patient information includes many details such as demographics, physiological measurements, lab test results, and so on. So to predict the diseases, two types of features are included for each patient. Time series and discrete features. And time series features are kind of physiological measurements such as heart disease, heart rate, blood pressure, and temperature, while discrete features are kind of symptoms, complications, and related procedures. And as a set of possible diseases, around kind of 400 diseases are extracted in a way to ensure that different target disease cohorts have no overlapping patients, and also that there are at least 20 labeled patients per disease. And prior information is given from the ICD hierarchy, with some categories kind of shown here on the slides. So for example, we can see that blindness and bacterial meningitis are both kind of nervous and sense come from the nervous and sense category. And the idea here is that kind of we can use these similarities, categorical similarities between diseases to potentially solve these disease prediction tasks given few labeled examples better. So experimental setup is similar to already considered applications, but kind of it's good to do the recap. So first, disease classes will be randomly sampled to define a task in each episode. And then given a task, we will sample disjoint support and query set. So in case of one-shot learning, support set consists of one positive and one negative example per class. And query set is used to compute the loss and update parameters. And then during model evaluation or testing, the model is evaluated on the three-way setting, which means kind of we will sample 3 random disease classes, and then we sample support set, again consisting of, so this is a typo again, one positive and one negative example per disease class, and then the performance is evaluated on the query set, which comes from the same set of disease classes as the support set. And considered baselines are optimization approaches MEML and mmeml, and metric-based approaches matching networks, prototypical networks, and relation networks. So we mentioned earlier, and PPN. So we mentioned earlier all these methods except mmeml, which is the extension of meml that introduces task-specific initializations, while PPN is another approach that considers graph relationships between the classes. And it learns kind of the propagated prototypes by accumulating the level-wise classification loss. And again, if you're interested in these methods, we provide the references at the end of the tutorial. So to evaluate the model's performance, tasks are generated using 2 different sampling strategies. So first one is the random sampling, and the second one is a subgraph sampling. So idea in the random sampling is that the few shot classes are randomly selected from the leaf nodes of the whole graph. While in subgraph sampling, the graph is divided into several subgraphs without overlapping target classes. And then each task is sampled from nodes within one subgraph. So which strategy leads to the harder task? The answer is subgraph sampling. Why? Because tasks are here constructed heterogeneously, and kind of tasks sampled from the same subgraph can be considered more similar to each other. So because subgraph sampling corresponds, for example, to the scenario which we need to distinguish between kind of very fine-grained classes, for example, Parkinson versus Alzheimer's disease. While in random sampling, that doesn't have to be necessarily the case. And the backbone network used, we have 2 types of features, time series features and discrete features. So the backbone network consists of one-directional convolutional neural networks for sequence structure learning, which handles time series features. And then there are discrete features, which are handled using a fully connected neural network. And then these representations are combined by learning by using one fully connected layer. And the combined features kind of produce the final patient representation. So let's now look at the results for three-way one-shot task and three-way five-shot task. So just as a reminder, three-way kind of means we are sampling 3 classes. One-shot means 1 support example per class, meaning 1 labeled example per class, and five-shot means 5 labeled examples per class. So first observation is that PPN and TADANet that leverage graph information significantly improve performance over the non-graph baselines. And this is very encouraging. And kind of this improvement is larger in the one-shot learning setting, which is intuitively expected because in the one-shot task, we have less information, so kind of background knowledge will have higher effect. And secondly, as expected, kind of subgraph sampling is a harder task, and we kind of see that the performance goes lower for the subgraph sampling. And finally, the final observation is kind of that there is still much space for the improvement because we can see that the performance is kind of far from satisfactory and from something that we can really use in practice. And next, let's examine how kind of does the weight regularization lambda affects performance. So to recap, lambda is kind of combining this initial embedding prototype and the final propagation prototype. And here on the graph, one-shot tasks for random and subgraph sampling are shown in the blue and orange color. So the graphs here, while the green and red color denote five-shot tasks. And we can see that kind of higher lambda leads to better performance, and improvements are much more apparent for one-shot tasks in both random and subgraph sampling. So in summary, these results show that if we have prior knowledge, either on the feature level or task level, we can expect to get better performance if we kind of incorporated it into our model. So the less number of labeled examples, kind of the more gains we can expect by incorporating prior knowledge. So far we covered kind of few-shot learning methods. We introduced the few-shot learning problem, explained the concept of metalearning, and covered 2 classes of these methods, optimization-based and non-parametric methods. We've seen applications of these methods for kind of drug discovery, cell response prediction, single cell annotation, and disease prediction. So if you want more, here are the references for kind of methods mentioned in this paper. And for the next part of the tutorial, kind of just to see where we are currently. So we covered all the few-shot learning topics. And next we will kind of cover open world learning and see kind of how can we learn to generalize without any labeled examples. Yeah, I can take a few questions.
So Henry asks, why is subgraph sampling considered? And is the conclusion that you should just use random task sampling? The idea, kind of the reason why you want to kind of check performance on the subgraph sampling is because it's more realistic. Because in reality, you would really need to kind of differentiate between diseases that are more similar, like because given a patient, you can be sure, for example, that you can easily maybe distinguish like between, I don't know, brain and lung disease, but it's much harder to distinguish with the fine-grained diseases. So when you think kind of about the real-world applications, the subgraph sampling is much more realistic use case. And Shaman asks, how do you combine the time series and the discrete features? Is it simply a concatenation or something?
Let me go back. So the idea is to kind of have one layer of fully connected neural network here, and to learn kind of to combine these features. You can try simply concatenating them, but if you have kind of enough examples, maybe you can get better generalization if you kind of learn to combine them. So this, I think, probably if you kind of need to combine different feature sets, it's good kind of to explore whether kind of which approach works better. And sometimes like simple concatenation will work very well.
Yeah, that's it.
Great. Thanks, Yusuf. So next topic we will cover in this tutorial is open world learning. I will start and then Jure will take over. So in this part of the tutorial, we will discuss how can we discover novel, never before seen classes. And we will then go a step further and see kind of how can we simultaneously like solve both tasks, kind of standard supervised task of recognize these classes seen during training, and also simultaneously discovering novel classes. And our learning goals are first to understand the concept of open world learning and basics of open world learning methods and kind of how can we implement them. And we will cover applications of these methods to single cell data sets on a cell type annotation task. So in conventional machine learning, we assume that test data will contain exactly same set of classes as the training data. And this is so-called closed world setting. And we also typically assume that we have kind of abundant labels for each of these classes. So in this toy example, let's say we want to kind of recognize elephant, cheetah, zebra, and rhino classes. And then what we expect during test is that we will again get only elephant, cheetah, zebra, and rhino class. But the question is what to do if this assumption is not satisfied. So when we think about the real world setting, we can easily imagine that we can encounter new classes during testing. So in our example, well, let's say we train our model to recognize elephant, cheetah, zebra, and rhino, but then during testing, we get frog or octopus pictures, and this is kind of class the model has never seen before. And this is the open world setting. So ideally, the model should be able to recognize these classes and not just to recognize that these are some outliers, but also to recognize that frog is different from the octopus. So kind of group similar examples, novel examples based on their similarity. And this setting is actually very realistic when applying machine learning models to solve problems in biology. So in biomedical domains, labeled data sets are often very difficult to obtain, and they require a lot of costly manual effort, expert knowledge to hand label all the classes before we can even use machine learning models. So for example, we might train the model to recognize 3 types diseases, shown here in the blue, orange, and violet color. And then when applying our model into the wild, patients can have rare disease types for which we were not able to obtain training data. So in this case, we need models that are able to recognize these novel classes. And in the next part of the tutorial, we will see how can we develop methods that can discover novel classes. So the problem we are considering is the following. So we are given a set of annotated data sets that correspond to different tasks and in which examples are abundantly labeled. And we are then given a new data set with unknown class in which many classes can be unique, meaning that we can not find them in any other previously annotated data set. And the task is to kind of label every sample with its class in this unannotated data set. For example, abundantly labeled data sets may kind of correspond to different tissues, such as pancreas, lung, and heart. So for example, we might, in the single cell data, we might kind of, there is maybe one experiment on the pancreas data, and it is already labeled, we have the data, it is easily accessible. And then we are given a new data set, which is kind of coming from the brain cell type. And many cell types here are unique. And the question is kind of, can we reuse this already previously labeled data sets to solve this task? And our task is kind of label every cell with its cell type in the unannotated experiment. And to solve this challenge, the key idea in the approach named Mars is to kind of reuse this old, already annotated data to learn to generalize to new, never before seen data set. And the idea is that we start with a set of previously annotated data sets, and these data sets can kind of come from different experimental conditions, such as different tissues. And then we feed to the learning algorithm both sets of previously annotated experiments, our data sets, and the new completely unannotated data set. And we use these data sets as a metadata set in our meta-learning sense. And these data sets are kind of used to learn how to learn. And the idea is kind of that the learning algorithm can reuse these previously annotated data sets to achieve better generalization on the unannotated data sets, even though this data set might have classes that are not seen during the on these annotated data sets. So the key idea of the MARS approach is to map all data points from both annotated and unannotated experiments into the joint low-dimensional embedding space in which examples should group according to their classes. So to achieve this, MARS learns a nonlinear mapping function f that maps examples from both annotated and unannotated data sets into the joint low-dimensional space. So this mapping function f will take our input vector and embed it into this low-dimensional embedding space. And in this low-dimensional embedding space, we want to enforce examples coming from the same classes to be close, while examples from different classes should be far away. And at the same time, MARS learns the class landmarks, that is kind of class representatives or kind of class centroids in this low-dimensional space. And two types of landmarks are learned: landmarks for annotated data sets, which are kind of marked as pentagons in different colors, and also landmarks for unannotated data set marked in the gray color, because we don't know their identity. And the objective function here is to kind of force samples from the same classes to be embedded close, while examples from different classes should be embedded far away. So originally, MARS considers heterogeneous single cell experiments as data sets, and then kind of cell type annotation task as the target data set. But we here made the approach more general, and in the application part, we will see the application to single cell genomics. So the way we can kind of design the desired objective function is that for annotated data sets, we want to have part of the objective that says that samples from the same classes should be embedded close to the ground truth landmark in the embedding space. And the way we can kind of mathematically achieve this is that our objective function, we want to kind of minimize the distance between a given data point and its kind of landmark. But because here data sets are annotated, we can easily compute the ground truth landmark. And the intuition is kind of the samples with the same class annotations should have similar embedding vector. For unannotated data set, we don't have the ground truth landmark, we don't have any annotations, and the idea here is to kind of learn both the landmarks and embeddings. And instead of kind of minimizing the distance to the ground truth landmark, we will minimize the distance to the closest landmark. So for each cell, we will find its closest landmark and then force cell close to its closest cell type landmark. And finally, for both annotated and unannotated data sets, we can kind of push the landmarks far apart from each other. And mathematically, this means that we want to maximize the distance between the landmarks. And in this case, kind of the intuition is basically that landmarks of different classes should have different embedding vectors. So let's now look how to mathematically formulate this, and let's define the notation first. So we will denote set of annotated experiments as di, and they will be given as xi as a set of our examples, and yi, which are class annotations. And then we are given an annotated dataset u, and our goal is to predict yu, meaning labels for this unannotated dataset. And we will denote embedding function as f theta as earlier, and it will kind of take the original m-dimensional vector and project it to the lower z-dimensional embedding space. And we will learn here both the landmarks for annotated data sets and landmarks for unannotated data sets, and they are marked as this lowercase p, where if p is indexed by I if these are landmarks for annotated experiments. And so in total, for annotated experiments, we need ki landmarks. So ki depends on the number of cell types in current annotated data set. So this depends if we kind of consider generalization, for example, across tissues, for different cell types, this will depend how many cell types per tissue you have. And each kind of experiment or each data set, you can think of it as one tissue. And recall that for each annotated experiment, we want to kind of force data points to be close to their ground truth landmarks, and also kind of separate landmarks to be far apart from each other. So let's kind of write this mathematically. So for each annotated dataset DI, we will first force samples close to their ground truth landmarks. So for each data point, we will look at the distance to the ground truth landmark in the embedding space defined by the f theta. And kind of we will obtain the ground truth landmarks by looking at the indicator function between the current class and the label example. So this indicator function will be 1 if the label of this current example is the k, meaning corresponds to the landmark of this, it corresponds to the class which this landmark is representing. So each data points will really take part in this sum only once, because this indicator function will be true only for one class, because these are annotated experiments and we already have the annotations for this part that we can reuse. And the sum will go over all examples and over all over ki, which is the total number of classes in this unannotated dataset, And then we want to kind of force the landmarks far apart from each other. So to achieve this, we will look at the pairwise distance between the landmarks they are in the embedding space. And we will, of course, sum over all the pairs and normalize by the total number of pairs. And the lambda is kind of the regularizer defining the strength of this part compared to this part. And the minus here is basically because we want to maximize this part compared to this part, which is minimized. And then for unannotated dataset, we will do the similar thing, but here we want to force cells close to their closest landmark, or cells or our learning examples. So in this case, this landmark, we will look at the distance between the embedding of the data point and the landmark of class K again, but instead of the indicator function, we will now have the minimum. And this minimum means kind of that we will check which the closest landmark for each data point. And we will again sum over all examples and over all the distances. So the total, again, each example, so the total number of examples we will have corresponds to the total number of examples, because each learning example will take part in the sum only once, because we will only find the minimal distance. And then we will force kind of landmarks far apart from each other again. And this means, again, that we will look at the distance between the landmarks and then sum over all the pairs. And again, normalize by the total number of pairs and regularize by lambda. So the final objective function consists of the sum of the losses of over all previously annotated experiments. And then there is a tau which regularizes importance of this part of the unannotated dataset. Originally, it was set to 1, but if you kind of want to enforce this part more, you can send to higher value. And then we minimize, the algorithm will optimize over landmarks and embedding parameters data. So in Mars, embedding function the backbone network or embedding function F used is a fully connected neural network, which consists of two layers, because the inputs are these are single cell. This is a single cell problem, as we said earlier. But again, you can replace the backbone depending on your input type. And before fine-tuning the network on the design objective function, network is first pre-trained using the autoencoder. So in autoencoder, we give the input our input vector, and we aim to kind of reconstruct the input vector at the output. So the idea is kind of input and output layers have the same dimension, but kind of these middle layers will have kind of much lower dimensionality. So the network is forced to learn to compress the data. And it's kind of forced also to learn the only essential latent features and kind of to ignore non-essential sources of variation. And it's shown kind of that this pre-training step serves as a good prior for the parameter space, and is kind of very useful to generalization to the unannotated dataset. And autoencoder pre-training is used in the original Mars paper, but this step can also be application dependent. For example, if you have kind of some auxiliary labels for your task, you might kind of reuse these auxiliary labels in order to kind of define different pre-training tasks. And after pre-training, we will remove the decoder part and use encoder weights to initialize the network. And we will also initialize the network by kind of running k-means clustering in the embedding space pre-trained with this network. And then we will fine-tune the network with the designed objective function. And during the inference, we will embed the data points from the unannotated data set in the learned shared embedding space. And then we will assign data points to the class of the closest landmark. And next, we will see how to apply these methods for cell type discovery in single-cell genomics, and Jure will tell you more about that. So are there any questions at this point? I can take one question, and I can also respond to others in the chat.
Yeah, I think there's one sort of high-level question. Is there any substantial difference between landmarks, prototypes, or concepts between these different models?
You can see them as in the same way, actually. So these are kind of your class representatives. So if you think of it kind of in the k-means clustering sense, this would be kind of just the mean. You will in k-means clustering, for example, you will just take the average of your original examples. In prototypical networks, you kind of take the average over your support set. But this is all kind of happening in the embedding space instead of the original space. But these are kind of, in all these examples, this kind of, you can see these prototypes, landmarks as kind of your good representatives of your learning examples.
Okay, great. I think the rest you can take in the chat. They're kind of specific.
Sounds great. Thank you, Yusuf.
Good. Thank you very much, Yusuf, and thank you, Maria. So I will take on the tutorial and talk about the application of the methodology that Maria presented to single cell data, cell type discovery in single cell genomics. And this is based on the paper published last year in Nature Methods. All the code and everything is for the method is also available. So here is the problem, right? We are, you can think that on the input, we are given this matrix where we get a gene expression sequence for each individual cell. So the number of rows in the matrix is the number of different cells we have sequenced. The number of columns is number of genes. Think of that as, order of 20,000 columns. And the entry of the matrix tells us the expression level of a given gene in a given individual cell. And of course, cells in the body are of different types. So we can think that perhaps some human pre-annotated some of the cells with a given cell type. So maybe the blue rows are the T cells, green rows are the B cells, and orange rows are the fibroblasts. And this is kind of what we would like to do. Of course, when manually annotating cell types is a very expensive, slow, high human intensity task. And the question is, how do we discover novel cell types across tissues, species, as well as different sequencing technologies? So basically technologies that measure gene expression level, perhaps without any annotations given to us ahead of time. So basically a completely unsupervised method. So let's see how we do this. So the data sets we will be using for this part come from the Tabula Muris and Tabula Muris Senis projects at Chan Zuckerberg Biohub. And over the time, more than 100,000 cells have been sequenced from 23 different tissues of a mouse. And here each cell is described by its gene expression profile. So each cell is a 23,000 dimensional vector. And here you see, this is the figure from the original paper. This is a low dimensional embedding, a 2-dimensional embedding of this 23,000 dimensional space where every dot is a cell. And here cells are colored according to their cell type. And you see how basically cells of a different cell type have a different gene expression profile, so they are in different parts of the space, but you can also see that somewhere there is a lot of overlap and a lot of mixing, and this is not perfect. So the question that we wanted to ask in this Mars method is, how do we generalize, how do we build machine learning methods that can learn on some existing data and then be able to generalize to unsealed cell types to new tissues? And here we are going to do the training and prediction, where we want to take this 23,000 dimensional gene expression vectors and reduce dimensionality to 100 dimensions, right? We want to have neural networks that learn this 100 dimensional embedding for every cell. And then given source tissues with annotations and target unannotated tissue, we'd like to do the following. We would like to train the model on this annotated source tissues, where we use tabula muris cell annotations on source tissues, but then we wanna use this model that we trained on, let's say, heart and lung, where the people have already annotated the cell types. We'd like to use this data to learn how to generalize to a new cell type that we haven't yet seen, that is unannotated, right? So the point is that during training time, we can use cell annotations from a given set of tissues, and then we would like the model to work and identify cell types on a brand new tissue that we never seen during training. So that's the task. Okay. What will be the baseline methods that we will compare against? One is the Louvain method, which is basically a clustering method, where basically for a given tissue you perform clustering of the cells, and whatever clusters of the cells you identify, so every cell is a 23,000 dimensional vector, you create some kind of nearest neighbor similarity network on top of this, and then Louvain is a community detection method, and you identify the clusters, and each clusters you say is a separate cell type. So that's a method that only uses clustering, doesn't use any annotations from other cell types. And then we will also compare to two more advanced methods. One is called Similar, which is an unsupervised similarity learning approach for clustering and dimensionality reduction, and it learns cell-to-cell similarity metric using multiple kernel learning. So again, a method that doesn't do any transfer of knowledge between annotated tissues and the target unannotated tissue. And then we'll also have the last method that is built on a hierarchical Bayesian model and the deep neural networks, where we pre-train deep neural network with the same data that we use to pre-train our approach, that I will, so the Mars approach. When we apply these different methods, here they are, 5 columns, 5 different methods, and then different tissues. You notice that our method outperforms the next best competitor by 45% relative, measured through the adjusted RAND index. And basically, we are able to outperform all other methods on essentially all tissues. And wherever the Mars is not the best method, it's super close, perhaps, except on lung, perhaps, to the top contender. And the setting here is the following: is that we say, let's identify cell types in aorta, and aorta is unannotated. We just know the cells. We don't know what cell types appear in aorta, but we can use all these other tissues which we assume are annotated and kind of learn the embedding space structure on all these other tissues, and then hope that we can better identify cell types on the aorta. And this shows that we are able to achieve, in some sense, this transfer from one set of tissues to a new unlabeled tissue to a very high success. This is one example. We also notice that when you pre-train, when you build your Mars model on more annotated tissues, the performance on this holdout unannotated tissue gets better. So here we say, what is the number of annotated tissues on which we train? And then we take this model and apply it to the unannotated tissue that is different than the tissues in the training data set, if you want to think of that, right? And it seems, and it shows that the more knowledge about the cells, the cell types you use in the training stage, the bigger the performance performance boost you obtain, which is awesome to see. Another interesting thing is that as we can examine these embeddings of Mars, and we can do a low dimensional projection, and you can see, for example, here are two different tissues, diaphragm and Mars, and you can see how basically cell types get very naturally separated out into separate clusters that are very clean and very far apart, right? Maybe you see kind of single outliers, which is unclear. Is this really a cell type of MS, and this endothelial cell kind of got mislabeled? But you see basically things very nicely spread apart. Another question that we'd like to ask in our analysis is, can we go beyond the cell type discovery and actually provide human understandable names to discover when we discover new cell types? And the way we achieve this is that we use these landmarks that Maria was talking about from annotated experiments to generate interpretable names for a discovered cell type, right? So the idea basically is that we can take our labeled tissues, we embed them, and for every cell type in the annotated tissue, we have a landmark. And then when we take the unannotated tissue, we take the cells and we project them back into the same space. But if we say, you know, what is the cell type for this particular bunch of cells here, we could say, hey, these cells are similar to whatever cell type this particular landmark here, this pentagon belongs to. And we could say, these orange cell types here are likely of the cell type that is characterized by this pentagon here, if you see my mouse pointer. And this way, we can basically give names and suggest what are most similar known cell types to this newly discovered cell types, right? So basically, we can probabilistically define cell type based on the distances between of the landmarks from the annotated experiments, right? So how do we estimate these probabilities is the question. How do we create a ranking list here? How do we kind of sort by the distance? And we estimate the distance by basically using kind of Gaussian modeling and using a probability density under a Gaussian distribution centered at the mean of a given cluster, right? So we say, what's the landmark, and then what is the distribution modeled by a Gaussian of other cells that belong to that landmark, and that allows us kind of to estimate the density of the cells around the landmark, whether they are more spread out or more concentrated, and this way estimate in some sense the probability, the likelihood, or the distance. You can think of it that way as well. To give you an example here, right? I showed you this before. Pentagons are the landmarks. Here is a new set of unannotated cells. We don't know what they are. We can zoom in, look at it, and we see that there are two landmarks very close to these cells that are unannotated yet. And then basically what we can do is identify what are the cell types of these two landmarks that are close to it. And if we look at it, these are actually satellite muscle cells, which turns out to be exactly the right classification, the right cell type for this unknown cell type. So that's what this allows us to do, and is a novel capability that other methods doesn't allow you to do. We are actually able to tell you what cell type this likely is, or what is it most similar to in this sense, right? So that is great. Here's another example. When we take these orange cells, we can say what are the closest landmarks, and here we say, with likelihood of 60.6446, it's a B cell. Maybe it is a T cell. A T cell landmark is also quite close, and with the remaining, it might be something else. So we are able to basically give these probabilities to names of unannotated cells. And the last example. These are the cells that get in this cluster. We see a lot of landmarks here. We actually know that these are the macrophage class type cells, but Mars does not know that. Mars says this is most likely macrophage, maybe it's something else. And the annotation is correct. We actually identify that all these cells belong to the same cell type, and we can also tell what this given cell type is. So that's the idea. What we can also do is to ask a question: what if we want to recognize previously seen and also discover new cell types simultaneously, right? The point being that in Mars, we basically we discover these clusters, but we don't, and we can assign them names, but it's hard for us to say, oh, this is brand new, we've never seen it, and this is something old that we have seen in some other tissue. So what I want to talk about next is this exciting area of open world semi-supervised learning. We have a paper in submission, and it's also available on archive for people to be able to read. So how does this relate to what we have learned so far? So far we talked about discovering cell types, discovering novel classes, where we pre-train and then learn to cluster by reusing previously annotated tissues. And the challenge is, what if we want to simultaneously discover new classes and assign examples to the previously seen classes, right? So basically the idea is I get a new tissue, whatever cell types are existing cell types, I want to I want to classify them into known cell types, and whatever is kind of a new cell type, I want to put on the side and say, here is a new cell type. So this is now a different, more challenging task. The way you can think of this is, if you think, let's say in terms of species classification, because it's easy to understand. In the supervised learning, we are given, training data. We say here are instances of, I don't know, octopuses, gepards, and elephants. And then, we are given some images, and we know that we are only given images from these three species, and we have to decide for each image which of these three classes it belongs to. Right, so basically we want to assign it to an elephant, octopus, or cheetah, and we know that it's one of the three classes, and we cannot see, I don't know, some other animal in the test set. In novel class discovery, the idea is the following: we are given some training data. Maybe we get the images of this labeled training species. But then at the test time, maybe some novel species arrive. And the problem of novel class discovery now is to say, these orcas are class 1, zebras are class 2, and frogs are class 3. But we just discover novel classes. We know that whatever we'll get in the test data does not come from the species we get at the training time. So this is novel class discovery. And what open world semi-supervised learning does, it combines the two, right? With the idea that at the training time, we get images annotated with species, and at the test time, some of the images might come from existing known species, and some images might come from, let's say, unknown species. So what the method has to do is to say, oh, I've seen, this kind of species I haven't seen yet, so I will call it new species number 1. I haven't seen this funny looking species, I'll call it species number 2. But this particular species I have already seen, and I know it's called a cheetah species, right? So at the same time, we want to classify images that we already know what class they belong to into a given class. And whatever is kind of novel, we want to discover a new class for those images. So in some sense, we want to simultaneously discover novel classes and also recognize previously seen classes. Or another way, we want to do clustering and classification at the same time, right? We want to classify into existing classes if it looks like this is something that we already know, or we want to cluster it into a novel cluster if it's a species that we have never seen before. So that's the idea. Why is it hard? Because novel classes from data can negatively affect the classification performance on the seen classes, because the classifier gets confused. Novel class discovery task is hard by itself because we have to basically do clustering and identify these novel classes. And why would you want to solve this task? Is because it's a very realistic setting, again, especially in biology and natural sciences, where we cannot expect to identify and label all cell types. We cannot expect and label all species and say this is the set of species, there's nothing else in the world. There may always be something new that we haven't seen before, and the machine learning method should be able to recognize that. So that's the key here. An example would be disease classification, right? Maybe on the training data, I have seen 3 different diseases and I build a classifier for that. But once I deploy the classifier to the real world, maybe some people will have these 3 known sets of diseases, but I also want to be able to discover people that have a disease that I've never seen. And I'll call this, unseen disease number 1, unseen disease number 2. While, the rest of the people will say, oh yes, this is the orange disease, this is the violet, and this is the blue disease. So that's kind of what we want to do at the same time, right? We want to train a classifier that associates patients to previously seen diseases, but also discover classes of previously unseen disease states. The method we are going to talk about is called ORCA. And ORCA is an approach that introduces additional classification heads for each newly discovered class. So basically, the way you can think of it is that we are going to train this neural network that is going to have one head per known class, and it's going also to have kind of unallocated heads for novel classes. And we are going to train this system such that if data comes from a seen class, the correct neuron will fire for that given class. And if the example comes from an unlabeled class, from an unknown class, some other neuron here for that given unlabeled class is going to fire up. So that's the idea. And of course, the question is, how do we achieve this way that basically we have a neural network that automatically activates some neurons depending on the structure of the data? The way we do this is that our loss function will be composed from 3 different terms. We are going to have a supervised loss function. This will be a cross entropy. We are also going to have a unsupervised objective that will work on the unlabeled data. And then we'll also have some regularization term that will kind of regularize our model towards what we call uniform distribution. So let's see how this works. So first, we'll talk about each of the losses in turn. So first, we want to talk about the unsupervised objective. In the unsupervised part of the objective function of the loss, we want to predict whether 2 examples belong to the same class, right? If we get 2 images of an octopus, I want to predict this is from the same class. And for label data, we can use existing labels, and this is, it's easy to set up. We know which examples of octopus are of octopus, and we know which pairs are from the same species and which are not, if the images are labeled. But of course, the question is, how do I get labels for unlabeled data? Right? If I have 2 unlabeled images, I don't know whether they come from the same species or not. Right? And the key idea here is that I'm going to generate what we call pseudo labels. Right? I'm going to kind of label these unlabeled images on the fly iteratively. And the idea will be that I want to find closest neighbors in the embedding space and regard them as instances of the same species, of the same novel class. So the idea is that I can take my images, I can embed them in the embedding space. And these 2 I know are of the same class because octopuses are pre-labeled, so I can put them together. While, for example, for Orca, Orca is unlabeled, but it's far away from the octopus, so I won't say these 2 are from the same class, right? So the idea is that images that get embedded close together, I'll say these are from the same class, and I will generate a pseudo label for them and just say, I think this should be from the same class, right? So this way we can create these noisy pseudo labels that we use as an objective in our training examples. And of course, as we are training and the embeddings change, these pseudo labels are also going to change, right? So that's the way we do it. And here is the formula for our unsupervised loss. It is the way we think of it is on the top is the input. We have our embedding multi-layer network, and once we have this embedding, we then have a classifier that predicts the class for a given label or for a given pair of images. And here we are using we are saying, we want the class of the z of the data point I and the class of the closest neighbor in the embedding points I prime. I want these two to be to be similar, right? I will take the dot product, I will multiply them, so the multiplication will be high only if the two values are high together, right? And that's the intuition here. So basically, we want data points that are close together, nearest neighbors, to belong to the same class. Now, for the supervised objective, this loss CE, as we call it, using standard cross entropy on loss on scene classes results in learning scene classes faster than novel classes, right? Kind of the scene classes, the labels are so strong, they have no noise, that that classifier learns super quickly, and there's nothing left for the unlabeled images, right? So we need to fix this, right? And we need to kind of make sure that both the labeled data and unlabeled data kind of provide the same level of signal, even though the labeled data is so clean that it overpowers the unlabeled data. The key idea to do this is that we have this uncertainty-based adaptive margin to avoid learning scene classes too fast. And the way we do this is essentially we have the following loss where what is new and different is this term mu hat, and this is our estimate of the uncertainty. And then we have the constant, the hyperparameter lambda, that is our regularizer defining the strength of or the scale of uncertainty. Of course, now the question is, what does this uncertainty mean, and how do we define it? And if you look at this, right, this will be now a normalized probability, and you can see that for a given class, we are basically adding in this uncertainty term. And the way we estimate this uncertainty term is that we estimate it from the confidence of unlabeled examples computed from the output of the classifier. So the intuition here is that at the beginning of the training, we have larger uncertainty, we have the larger margin, so we have larger intra-class variance between the scene classes. And as the training kind of converges and continues, we have a smaller uncertainty, margin gets to 0, and we are approaching kind of more the standard cross entropy loss, where we have small intra-class variance, right? Where basically our predictions are getting more and more narrow, more and more certain. And this actually allows us now to train a classifier that can recognize existing classes and also at the same time discover novel classes. And I'm going to show you this on a cell type annotation task from the codex data. And this is an ongoing work, a collaboration with Mike Snyder and Gary Nolan's lab at Stanford University. So let me tell you about this. This is still work in progress, right? So Codex is a new exciting technology that allows us to do multiplexed imaging data of cells. And here's an example of this imaging of some region in kidney, right? And it provides us full spatial context, right? So these are individual cells, and we know how they are organized and where in space they are. It is a high throughput it gives us high throughput single cell information, but it only gives us about 50 protein targets within a single tissue section, right? So we don't get all the 23,000 tissue genes, but only about 50, right? And the question here is, how do we accurately identify cell types where we have both spatial information available as well as the gene expressions of these 50 protein targets, and how do we generalize across different tissues and patients? Here in this codex, in this data, we actually have human data, human tissue data. So the challenge here is the challenges here compared to RNA-seq type data that we discussed in Mars. We get additional kind of sources of noise from imperfect segmentation of cells, and also imaging artifacts and issues with image processing. Again, we only get 50 proteins per cell, not 23,000. And we also have the spatial context, the spatial locations. As I said, the data sets are coming from human small intestine and colon, generated by the Human Biomolecular Atlas program, or HubMap program, that is run by Stanford University. For validation, we use quantitative validation using human expert labeled ground truth information of 4 regions of small intestine with ground truth annotations, as well as expert annotations across different donors. The setting is that we are going to leave out one region of a tissue, or leave out one patient, one donor, one human, and try to learn on other humans. So here is cross subject transfer, while in blue it's a cross region of the same tissue transfer. So ORCA assigns cells to previously labeled cell types and discovers new cell types. As I said, basically in ORCA we have a set of prediction heads. The first set of prediction heads is reserved for the scene classes, and then our objective learns to fire unassigned prediction heads for novel classes. And here the backbone will be a simple 2-layer fully connected neural network. And interestingly, if we apply our ORCA approach to leave one region out validation, where we find that ORCA outperforms other methods and agrees much more strongly with human annotations. So the agreement is 76%, rather than, for example, Louvain is only 38%. So it's basically a factor of 2 improvement, which is quite stellar. And here is the data for leave one donor out example, where we try to transfer across different humans, different donors. And here I'm showing you for different cell types and for given marker genes, you can see how basically the cell types discovered by the method have this highly expressed marker genes that are actually correctly identified. And when we look at the spatial structure of that tissue in different and here dots are cells, and cells are colored by different colors based on their cell type assigned by ORCA. You see now we can really clearly see this kind of organization of a tissue, organization of a colon, right? This is a colon in our case, kind of a section, like a cut through the colon is shown here, right? So in this open world learning, a method that I briefly overviewed called ORCA allows us to discover existing classes as well as identify novel classes. So basically identify novel clusters of data. What we have covered is we talked about how to generalize to unseen classes and transfer the knowledge across different biological contexts. We talked about novel class discovery methods, MARS, and then we also talked about a more ambitious method called ORCA that works in this open world semi-supervised learning. And I talked about the application to single cell annotation using RNA-seq type data. And we also talked about codex annotation type data. Single cell was on mouse mainly, and we also did something on fly. Codex is on humans. So if you are interested and you want to know more, here are links to papers, the Mars paper, the ORCA paper. Both of these two papers also come with code that is available on our group's repository. This code is freely available. You can download and it's ready to use. If you have any questions or any feedback on the code, we would be very excited to hear from you and support you in using our code. And we also have a website where we basically give also all the pre-processed data code so that all our experiments in the paper can be replicated so that the code can be stress tested and so on. So with this, I am done with this part of the tutorial, and Maria is going to take over.
So maybe Jure, you can answer the question, have you explored or plan to explore these methods on multi-omics data, for example, transcriptomics or proteomics?
That is a very interesting question, and especially the ORCA method is very much ready for this, because even if you think of the codex in the codex data is also kind of this multi-modal in a different way, right? We have the expression profile plus spatial locations. So extensions to multi-omics is very natural, and we are very excited for this direction to be explored. Yeah, thank you for the question.
Thank you. So I will now take over with the concluding remarks. So in the remaining time of the tutorial, we will provide kind of useful practical advices for training the models, give guidelines on available resources and libraries, and outline future directions kind of for applying meta-learning models in biology and biomedicine. And we also prepared a demo example of applying prototypical networks for cross-tissue cell type annotation task that can kind of help you to get started easily. So let's first start with kind of some practical tips for training the models. So first, data pre-processing steps such as data normalization, feature scaling are kind of always essential steps when building machine learning models. And verifying and understanding your data is extremely important. Kind of you can compute some basic statistics, visualize the data, and kind of, because regardless how good your model is, if you feed the garbage in, you will get the garbage out. So kind of it is super important to never underestimate the importance of this step. Regarding the kind of optimizer, the most popular optimizer to use is Adam, which automatically decays the learning rate and computes adaptive learning rate for each parameter. It is fast and efficient and kind of tends to work well for most of the problems. Regarding architecture, you usually don't need more than 4 layers, and kind of the ReLU activation function is usually a good choice. And it simply kind of thresholds a matrix of activation at 0, and it speeds up the convergence. Another tip is to use batch normalization. In batch normalization, kind of normalizes a layer input by you subtract the mean of the mini batch and divide by the variance of the mini batch, standard deviation of the mini batch. And through kind of this batch normalization, we can ensure that the input for every layer is distributed around the same mean and standard deviation. And it allows you kind of to have higher learning rates and speeds up the training process, and also makes a network more robust to initialization. And you usually apply it after a fully connected or convolutional layer and before a nonlinearity. So how to prevent overfitting? So one regularization strategy is that you can use dropout. So the idea in dropout is kind of do randomly drop out neurons during training with some probability. And that forces the model to kind of avoid relying too much on like a particular set of features, because it is forced to also explore other features. Another strategy to avoid pre-training is weight regularization. Kind of this ensures that the weights are not too large, and it usually boils down to incorporating L2 or L1 regularization in the objective function. Plotting the training and validation curves during training process will help you to decide when to stop training. So the point at which validation error starts to increase, although training error is still decreasing, is usually the good place to stop training. And this is so-called early stopping strategy, and it prevents overfitting, since higher validation error indicates the model starts to lose its generalization ability. So in this example here, you can see that the training error still goes down, but at some point validation error goes up. So it means kind of the model stops generalizing well. So this is a good place to stop training. When debugging a model, it is kind of often useful also to try to overfit a small batch. Because your network should be able to overfit a small batch. And if it's not the case, there is some issue with the architecture. Either the model is too complex or it is not complex enough even to overfit on a small set of examples. One advice, particularly for low data regime tasks, is that pre-training is often helpful. So this is especially the case for open world learning. And how to pre-train your model. So if you have an auxiliary task for which the labels are easily available, then you can train the model on that auxiliary task first. And this is often called self-supervised learning in the literature, and kind of the most popular approach is to have contrastive loss, which means kind of to learn representations such that similar samples stay close to each other, while these similar will be far apart. So in this case, you see that you need auxiliary labels because you need to kind of define which data points are similar or dissimilar to each other. And if you do not have auxiliary task, one approach is kind of to pre-train the network using autoencoder by trying to reconstruct input at the output of the network. And in this case, during fine-tuning, what you will do is kind of remove the decoder part of the network and use only pre-trained encoder. And there are three ways for leveraging pre-trained weights and fine-tuning the network. So first approach is kind of you fine-tune only last layer and freeze all others. The second approach is that you fine-tune few last layers and freeze others. And finally, you could kind of fine-tune the whole network. So which strategy to use? This depends how much label data you have. So in general, the more label data you have, you can allow fine-tuning more layers. And in Fusha tasks, where you only usually have few labeled examples, it is usually the best to only fine-tune last layers. And if you do not have any prior experience with deep learning library, kind of the good place to start is following the tutorials in some of the deep learning libraries, such as PyTorch, TensorFlow, or Keras. PyTorch and TensorFlow are the most popular deep learning libraries. Keras is kind of more high level. So if you just want to apply deep learning method to your problem, then Keras is usually a good way to start. But if you want to kind of explore these methods, if you want to kind of extend existing deep learning methods, you would need to go with PyTorch or TensorFlow. Where to get the data to start with? So some data resources, for example, which we covered in this tutorial, are listed here on the slide. For example, at our SNAP Stanford website, you can find mouse cell atlas data for a cross-tissue cell type discovery task. And we also use this data for the demo example. We also put together kind of smaller single cell benchmark data set for cross-species and cross-technology transfer. They are available at the SNAP Stanford EduMars. And then kind of data for drug response prediction task covered in the first part of the tutorial is available at the project GitHub page, also shown on the slides. And if you want to use the Mimic3 dataset, which we covered for the disease prediction task, this dataset is freely available, but it requires few steps before getting the data, such as kind of signing the data use agreement. But there is a smaller demo example, which is immediately available. So kind of it's good to explore this demo example. The link is here to first see whether this dataset is the right dataset for you, and then you can kind of request the access for the whole dataset if you want to go forward. And we've seen that incorporating prior knowledge into the model can kind of help to generalize better in low data regime tasks. And prior knowledge is application dependent, but we provide the links to some kind of resources that can serve as a good starting point, such as gene ontology, cell ontology, and disease ontology that we were mentioning, and that are covered in this tutorial. If you use gene ontology, the great tool is Jure tools, which is a kind of Python library, which makes the use of gene ontology very easy. And the best, probably the best place, if you are not sure like which ontology you want to use, the best place to start is kind of Obo Foundry, where there are many ontologies kind of listed for biomedical data, and you might kind of find the right prior knowledge you need there. We also cover different few-shot learning and open world learning methods such as optimization-based, MML, non-parametric, matching networks and prototypical networks, prior knowledge in haste methods, COMET and then AM3, open world learning method Mars. And here are the link to GitHub repositories of these methods that can help you to get started. All these methods are either originally written in PyTorch, or we provide a PyTorch link here, except of AM3, which only has TensorFlow implementation. And here we show kind of simple usage examples of the open world cell type annotation tool Mars. So you can simply create an instance of the Mars class, and as a parameter, you can provide a set of list of previously annotated experiments, your unannotated experiment, and the expected number of classes. Then you kind of train the model, and at the output you will get the landmarks and embeddings and annotations in the nData object, which is kind of the standard ScanPy library for Python. And the format is very similar to the pandas data frame. And the examples and the notebook usage are available at this GitHub link, which kind of leads you leads you through every step. And also to help you get started, we prepared a notebook which kind of shows how can you use prototypical networks for cross-tissue cell type annotation task on mouse single cell data. And here the model needs to kind of generalize from few labeled examples across different tissues. And the dataset and example are available at the tutorial website. And finally, let's outline kind of some of the future directions for applying meta-learning models in biology and biomedicine. So far, there have been very few applications of meta-learning methods in biology and biomedicine, and most of them we covered in this tutorial. But data sets with small number of labeled examples are ubiquitous in biomedicine due to huge manual effort, which we need to annotate this data, and also our limited knowledge of complex biology. So there is a huge opportunity to kind of broaden the range of applications of these methods and to use them to kind of solve challenging and important problems in biology. And interestingly, recent work has used kind of meta-learning to predict cancer outcomes across different cancer sites. And what they found is that not only just that the meta-learning helps for this task, but it also requires a magnitude less training examples compared to direct training. So for example, on this lung cancer dataset, meta-learning is trained using only 20 labels examples, and it achieves significantly better performance than using 250 labeled examples and fine-tuning the model. So this means that kind of instead of trying to collect more data, which is often extremely hard, we can also opt for a better model and maybe achieve similar or even higher performance. And although scarcely labeled datasets are prevalent in biology, there is also a huge opportunity of using the abundantly labeled datasets that might be related to the task you are trying to solve. And these abundantly labeled datasets can kind of be used for training meta-learning algorithms in order to learn to generalize to tasks that are scarcely labeled. So to discover new biological insights and really solve key problems in biology, we need to generalize to unseen classes across different biological contexts, such as different tissues, diseases, species. And meta-learning approaches are really a promising next generation methods that can enable new biomedical discoveries. So to get started with these methods, all we need basically is a metadata set of different but related tasks, and of course, new data set for your target problem, which you probably already have. And one challenge that might arise when applying meta-learning in more real-world applications is how to validate the model and how to be sure that the predictions are correct. And one direction that could enable broader use of meta-learning methods in biology and biomedicine is building interpretable methods. So far, there have been very few explainable few-shot learning methods, and the recent progress in the field has been mostly made using black box models. But how can we build the trust in a model whose predictions are based only on a small number of labeled examples, or extremely just one or none labeled example? And this is the essential question towards applying these methods in kind of real world applications, in which our decisions can have fatal outcomes. And while applying methods for kind of post hoc interpretations is a possible approach, recent works demonstrate the issues with this approach and kind of advocate that we really build inherently interpretable methods. So in summary, in this tutorial, we cover two types of missing data problems, few-shot learning and open world learning. We started with few-shot learning methods and explained the concept of few-shot learning and meta-learning, and covered optimization-based and non-parametric methods. And we've seen that incorporating prior knowledge into these methods can further help us to improve performance and can also be used to build interpretable methods. And we covered these diverse applications from like drug disease response prediction, drug discovery, cell type annotation, and disease prediction. And we explained the concept of open world learning, and we learned kind of how can we develop methods that can generalize to novel, never before seen classes. We covered methods for discovering novel classes such as Mars, and also methods for open world semi-supervised learning, which is even harder task in which we need to simultaneously discover novel, but also recognize previously seen classes. And we covered applications of these methods for annotating cell types in single cell RNA-seq data, and also for codex spatial imaging data. And thank you so much for your attention, and we hope that you found the tutorial useful and also are ready to use these methods in your own applications. And please take some time to fill out the tutorial survey, which is provided by the conference organizers. And you can find all the slides and demo example at our website. I can also go now, if you want to stay, briefly over our demo example for those of you if you're interested in. Thank you.